Hello there, and welcome to Midnight Sleep Science. Tonight, you're stepping into a world beneath the waves, where something extraordinary has been happening for over 200 million years. Coral reefs, often called the rainforests of the sea, are not forests at all, but intricate living structures built by millions of tiny animals no larger than a grain of rice. These animals, called coral polyps, are simple in their anatomy, yet astonishing in what they create. Together, they construct vast underwater landscapes that shelter a quarter of all marine life on Earth, even though coral reefs cover less than 1% of the ocean floor. What makes them even more remarkable is the quiet partnership at the heart of their survival, a relationship between animal and algae so seamless that neither could thrive without the other. In the stillness of warm, shallow seas, these reefs pulse with color, movement, and interconnected life. They are ancient, delicate, and endlessly generous, and they hold lessons about cooperation, patience, and resilience that reach far beyond the ocean. If you enjoy these gentle journeys, feel free to like and subscribe. But for now, let's begin. Beneath warm tropical waters, something small is happening that will change the shape of the ocean floor. A creature no larger than the tip of your finger is building. It does not think about what it is creating. It does not plan. Yet through its simple existence, it is laying down stone molecule by molecule in a process that will continue for thousands of years. This creature is a coral polyp, and it is an animal. Not a plant, not a rock, but a living organism with a body, a mouth, and a purpose. For many people who encounter coral reefs for the first time, this is the most surprising fact, that the vibrant structures stretching across the seafloor are built by animals, not minerals deposited by the sea. Each polyp is soft-bodied, translucent, and vulnerable. It belongs to the phylum Cnidaria, the same ancient lineage that includes jellyfish and sea anemones. And like its relatives, the coral polyp is simple in design but elegant in function. Imagine a hollow cylinder, open at one end. At the top of this cylinder is a mouth, a single opening that serves as both entrance and exit for everything the polyp consumes and expels. There are no separate systems for eating and excreting. The mouth handles both. Around this opening, arranged in a circle, are tentacles. These tentacles are slender, flexible, and covered in specialized cells. During the day, many coral polyps retract their tentacles, pulling them inward to rest. But at night they extend. They reach outward into the current, waiting. What they are waiting for is food. The tentacles are not merely sensing organs. They are weapons. Studded along their length are tiny stinging cells called nematocysts. These cells are among the most remarkable structures in the animal kingdom. Each nematocyst contains a coiled, thread-like harpoon. When triggered by the touch of prey, a copepod, a larval fish, a fragment of drifting plankton, the nematocyst fires. The coiled thread explodes outward in less than a millisecond, one of the fastest movements in nature. It pierces the prey's body and injects venom. The prey is stunned, sometimes killed instantly. Then the tentacles contract. They draw the immobilized prey toward the mouth. The polyp does not chew. It simply pulls the food inside. Once inside, the prey enters a cavity called the gastrovascular cavity. This is the polyp's stomach, digestive system, and circulatory system all in one. The walls of this cavity secrete digestive enzymes that break down the prey's tissues. Nutrients are absorbed directly through the cavity walls and distributed throughout the polyp's body. What cannot be digested is expelled back out through the mouth. The entire body of the polyp is organized around this central cavity. The polyp's outer layer, called the epidermis, forms a protective skin between the epidermis and the inner layer. The gastrodermis is a thin, jelly-like substance called mesoglea. This mesoglea provides structure without weight, a kind of biological scaffolding that keeps the polyp's shape. The polyp has no brain, no heart, no lungs. It does not need them. Oxygen diffuses through its tissues from the surrounding water. Signals travel through a simple nerve net. The polyp responds to touch, to light, to chemical cues in water. But what makes coral polyps truly extraordinary is not their anatomy alone. It is what they do with their bodies. At the base of the polyp, beneath its soft tissues, something hard is forming. The polyp is secreting calcium carbonate. It draws calcium and carbonate ions from the seawater around it and combines them into a crystalline structure called aragonite. This aragonite accumulates beneath the polyp, forming a cup-shaped skeleton called the corallite. The polyp sits inside this cup, protected by the stone it has made. This process is called calcification, and it happens continuously day and night as long as the polyp is alive. The polyp does not build alone. Coral polyps are colonial animals. After a polyp settles on a hard surface, a rock, a dead shell, a skeleton of another coral, it begins to bud. It produces a genetically identical copy of itself, attached side by side. This new polyp buds as well. And so the colony grows. Dozens of polyps become hundreds, then thousands, then millions. Each polyp continues to secrete its own calcium carbonate skeleton, and these skeletons fuse together into a single continuous structure. The polyps remain connected. Between them runs a network of tubes called gastrovascular canals. Through these canals, nutrients and chemical signals flow from one polyp to another. The colony behaves not as a collection of individuals, but as a single organism. When one polyp captures food, the nutrients may be shared with polyps that have caught nothing. When one polyp is injured, signals travel through the network, alerting the rest. Above the skeleton, surrounding and linking all the polyps, is a layer of living tissue called the coenenchyme. This tissue is the colony's connective fabric. It holds the polyps together and facilitates communication across the entire structure. And beneath it all, growing slowly, is the skeleton. Year after year, the colony adds layers. The older polyps, the ones at the base, may die, but their skeletons remain. New polyps settle on top of the old, building upward and outward. The structure expands. What began as a single polyp, no bigger than a grain of rice, becomes a boulder, then a formation the size of a car, then a structure larger than a house. Some coral colonies are hundreds of years old. Some are more than 1,000. The Great Barrier Reef, the largest coral reef system on Earth, has been growing for approximately 500,000 years. It stretches over 2,300 kilometers along the coast of Australia, visible from space. And it was built, piece by piece, by animals no larger than your thumbnail. The process is slow beyond human comprehension. A healthy coral colony might grow 1 to 3 centimeters horizontally each year. Vertically, growth can range from 1 centimeter to 25 centimeters, depending on the species and conditions. Some corals grow as branching structures, reaching upward like the limbs of trees. Others spread outward and flat, table-like formations. Still others form massive domes or boulders, adding layer upon layer in concentric rings like the growth of wood. Each colony has its own architecture. Each species builds according to its own genetic blueprint. But all of them are engaged in the same fundamental act, converting the chemistry of seawater into solid stone. The calcium carbonate they produce is not uniform. It begins as amorphous particles, loosely organized clusters of calcium and carbonate ions. These particles are transported to the site of calcification beneath the polyp's basal disc. There, they crystallize into aragonite, a specific form of calcium carbonate with a needle-like structure. The aragonite crystals interlock, forming a matrix that is both strong and porous. This porosity allows for the circulation of water and nutrients through the skeleton, even as it supports the weight of the colony above. The process requires energy. It requires materials. And it requires light. Coral polyps can capture food with their tentacles, but this alone is not enough to fuel the construction of a reef. The amount of energy required to extract calcium and carbonate from seawater, to organize it into crystals, to build layer upon layer of stone, this demand exceeds what the polyp can obtain through predation alone. The polyp needs another source of energy, a more reliable one, a source that never stops. This is where the story of coral reefs becomes something more than the story of a single animal. This is where cooperation enters. Because inside the tissues of the coral polyp and the translucent cells of the gastrodermis live billions of microscopic partners. They are single-celled algae, golden brown in color, photosynthetic in nature. They are called zooxanthellae. And without them, coral reefs as we know them would not exist. But that relationship will come later. For now, what matters is understanding what the polyp is: animal, builder, and foundation. Not all corals build reefs. Some corals are solitary, living alone as single polyps. Others form small colonies, but do not produce enough calcium carbonate to contribute to reef formation. The corals that do build reefs are called hermatypic corals, or stony corals. They belong primarily to the order Scleractinia, and they share certain traits. They live in warm water. They require sunlight. They grow slowly but persistently, and they form colonies. The colony is not merely a cluster of individuals. It is a superorganism. The polyps are genetically identical, clones produced through asexual budding. They are physically connected through the choanenchyme and the gastrovascular canals. They share resources. They coordinate their behavior. When night falls and feeding begins, the entire colony extends its tentacles in near unison. When danger approaches, a predator, a surge of sediment, the polyps retract together. Some colonies can live for centuries. The oldest known coral colonies are over 4,000 years old. These ancient corals have witnessed the rise and fall of human civilizations. They were alive when the pyramids were built. They were growing when Rome was founded. And they continue to grow today, layer by patient layer. But individual polyps within a colony do not live for centuries. They may live for months, perhaps a few years. When a polyp dies, its skeleton remains. New polyps settle on top of it, building upward. The colony persists even as its individual members are replaced. It is the skeleton, the accumulated calcium carbonate, that endures. This is how islands are built. This is how entire ecosystems are constructed. From the bodies of animals too small to see clearly with the naked eye. From the patient secretion of stone. From the unthinking cooperation of millions of polyps working in biological synchrony. Coral reefs can only grow in shallow water, generally no deeper than 150 meters. Below that depth, there is not enough light. And light, as we will see, is essential. The reefs grow upward toward the surface, pacing the sea level. If the sea rises too quickly, the reef may drown, unable to grow fast enough to stay within the sunlit zone. If the sea falls, the reef may be exposed to air and die. Most of the coral reefs alive today began forming after the last ice age, when glaciers melted and sea levels rose. As the ocean flooded the continental shelves, coral larvae found new surfaces to colonize. They settled on submerged hillsides, on rocky platforms, on the remains of older, drowned reefs, and they began to build. The Great Barrier Reef in its modern form is between 6,000 and 8,000 years old. It formed during a period of relatively stable sea levels. Before that, during the Ice Age 18,000 years ago, the sea level was 120 meters lower than it is today. The continental shelf where the reef now grows was dry land. There was no reef. Only when the ice melted and the water returned did the corals colonize the newly submerged terrain. This is a story told in layers of stone. Each layer is a record of the polyps that lived and died. Each structure is a monument to time, cooperation, and the quiet genius of calcification. The coral polyp, in its simplicity, accomplishes something extraordinary. It transforms the dissolved minerals of seawater into permanent structures. It creates habitat where none existed. It builds foundations that will outlast its own life by thousands of years. Consider the scale of what these animals create. Coral reefs cover less than 1% of the ocean floor. yet they support an estimated 25% of all marine life. More than 4,000 species of fish depend on coral reefs at some point in their lives. The actual number of species living on reefs, when you count not just fish, but mollusks, crustaceans, sponges, worms, echinoderms, and countless microscopic organisms, may exceed 1 million. Some scientists estimate the true biodiversity of coral reefs could be as high as 2 million species worldwide. Others suggest even higher numbers. The challenge is that many reef organisms are small, cryptic, and difficult to identify. A single square meter of reef can contain hundreds of species that have never been formally described by science. The deeper you look, the more life you find. The richest coral reefs on Earth are found in the Coral Triangle, a region spanning parts of Indonesia, Malaysia, the Philippines, Papua New Guinea, Timor-Leste, and the Solomon Islands. This area contains 76% of the world's coral species, more than 600 species of reef-building corals. It is home to 37% of the world's reef fish species, over 2,000 species swimming through waters that cover only 1.6% of the ocean's surface. All of this diversity, all of these thousands of species, depends on structures built by polyps. The reef provides shelter, feeding grounds, nursery habitat, and breeding sites. It creates three-dimensional complexity in what would otherwise be open water or barren seafloor. Fish hide in the crevices. Shrimp and crabs make homes in the branches. Octopuses den in the hollows. Sea turtles rest on the slopes. Sharks patrol the edges. The reef is not a static structure. It is constantly changing. Corals grow and die. Storms break branches. Parrotfish scrape algae from the surface, grinding away bits of skeleton in the process. Boring sponges and clams excavate tunnels through the calcium carbonate. The reef is being built and eroded simultaneously. What remains is the net result of construction outpacing destruction. When construction slows, when the polyps stop building or die, the reef begins to crumble. Wave action breaks it apart. Bioeroders hollow it out. Within decades, a dead reef can collapse into rubble. The structure that took thousands of years to build can be reduced to fragments in a human lifetime. This is why the polyp's continuous work matters. The reef exists only because polyps are actively secreting calcium carbonate faster than the forces of erosion can remove it. The reef is not a monument to the past. It is a living system, sustained by the ongoing metabolism of millions of tiny animals. But the polyp, for all its building prowess, cannot do this work alone. The energy required to extract calcium from seawater, to organize it into crystals, to maintain the colony, to reproduce, this exceeds what the polyp can capture with its tentacles. Zooplankton drift through the water column at night, and the polyps catch what they can. But it is not enough. The polyp needs more. It needs a source of energy that is constant, reliable, and abundant. It needs something that can convert the endless sunlight falling on the shallow seas into usable fuel. It needs a partner. Inside the tissues of nearly every reef-building coral, living within the cells of the gastrodermis, are billions of these partners. They are microscopic. They are photosynthetic. And they are the reason coral reefs exist in the form we know. This partnership, quiet and ancient, is where the story deepens. If you were to look at a living coral polyp under magnification, you would notice something unusual. The translucent tissue of the polyp is not empty. It is filled with color, golden brown, olive green, sometimes reddish or pale yellow. These colors do not come from the polyp itself. The polyp is transparent, a ghostly structure of tissue and calcium. The colors come from something living inside it. Billions of microscopic cells, each one too small to see without a microscope, reside within the gastrodermal tissue of the coral polyp. They are single-celled algae, photosynthetic organisms belonging to a group called dinoflagellates. Most of them belong to the genus Symbiodinium, though recent research has revealed many different species and genetic types. Collectively, they are known as zooxanthellae. The name means yellow animals, a misnomer from early scientists who mistook these algae for animal cells. But they're not animals. They are photosynthetic. They contain chlorophyll and other pigments that capture sunlight and convert it into chemical energy. And they live inside the coral in a relationship so tightly woven that neither organism can thrive without the other. This is symbiosis, not merely coexistence, but mutual dependence. The coral provides the algae with a safe place to live, protected from the currents and predators of the open water. It provides nutrients, nitrogen in the form of ammonia, phosphorus from the polyp's metabolic waste. These are the building blocks the algae need to grow and reproduce. The coral's tissues are transparent, allowing sunlight to penetrate and reach the algae within. In return, the algae perform a service that makes coral reefs possible. They photosynthesize. Photosynthesis is the process by which plants and algae convert light energy into chemical energy. Using sunlight, water, and carbon dioxide, they produce glucose and oxygen. The glucose is a sugar, a form of stored energy that can be used to fuel cellular processes. The oxygen is released as a byproduct. But in the case of zooxanthellae living inside coral tissue, the glucose does not stay with the algae. Most of it is transferred to the coral. The algae release up to 90% of the organic compounds they produce through photosynthesis directly into the coral cells. This includes glucose, glycerol, and amino acids, the raw materials the coral uses to build proteins, fats, and carbohydrates. This transfer is not passive. It is an active metabolic exchange. The coral and the algae are so deeply integrated that their biochemical processes are intertwined. The waste products of one become the nutrients of the other. The coral's ammonia feeds the algae. The algae's sugars feed the coral. It is a closed loop, efficient and elegant. The amount of energy the coral receives from its zooxanthellae is staggering. Studies have shown that photosynthesis by zooxanthellae can provide 80 to 90% of the coral's energy needs. This means that the vast majority of the fuel required to build the reef, to secrete calcium carbonate, to grow and reproduce, comes not from the polyp's tentacles capturing plankton, but from sunlight transformed into sugar by microscopic algae. Without this partnership, reef-building corals could not exist in the form we know. The energy available from predation alone would not be sufficient to support the metabolic demands of calcification. The polyps would grow more slowly, if at all. The massive structures of the reef would not form. Coral reefs as ecosystems would not exist. This is why reef-building corals are found almost exclusively in shallow, sunlit waters. The zooxanthellae need light to photosynthesize. Below about 150 meters, there is not enough light penetration to support photosynthesis. Without photosynthesis, the algae cannot produce sugars. Without sugars, the coral cannot thrive. So the reef stops growing. The relationship between coral and zooxanthellae is ancient. Fossil evidence suggests that corals and dinoflagellate algae have been living together for at least 200 million years, since the Triassic period. Over this immense span of time, the partnership has been refined through evolution. The coral has adapted to house the algae. The algae have adapted to transfer their photosynthetic products to the coral. The two organisms have become so interdependent that they function, in many ways, as a single entity. But this partnership is not without its vulnerabilities. The balance between coral and algae is delicate. It depends on stable environmental conditions, consistent water temperature, adequate light, low nutrient levels in the surrounding seawater. When these conditions shift, the partnership can break down. The zooxanthellae reside inside specialized compartments within the coral cells. They are not floating freely in the tissue. Each algal cell is enclosed in a membrane-bound structure called a symbiosome. This keeps the algae separate from the coral's cellular machinery while still allowing the exchange of nutrients and waste products across the membrane. The number of zooxanthellae in a coral colony is immense. A single square centimeter of coral tissue can contain more than 1 million algal cells. In a large coral colony spanning several meters, The total population of zooxanthellae can number in the trillions. These algae are not evenly distributed throughout the polyp's body. They are concentrated in the gastrodermis, the inner tissue layer, where light can reach them. During the day, when sunlight penetrates the water, the zooxanthellae are active. Their chloroplasts capture photons of light. The energy from these photons drives the chemical reactions of photosynthesis. Carbon dioxide, dissolved in the seawater and taken up by the coral, is combined with water to produce glucose. Oxygen is released as a byproduct, some of which the coral uses for its own respiration. The glucose produced by the zooxanthellae is immediately available to the coral. The coral uses it to fuel its metabolism, to grow, to repair damaged tissue, to reproduce. But critically, the coral also uses this energy to power the process of calcification. The secretion of calcium carbonate requires energy to extract calcium and carbonate ions from seawater and organize them into crystalline structures. Photosynthesis by zooxanthellae provides the fuel for this process. In fact, calcification rates in corals with zooxanthellae are significantly higher during the day than at night. This suggests that photosynthesis directly enhances the coral's ability to build its skeleton. The exact mechanism is still being studied, but it appears that the removal of carbon dioxide by photosynthesis shifts the chemical balance in the coral's tissues, making it easier for calcium carbonate to precipitate. Additionally, the energy provided by the algae allows the coral to actively pump calcium ions to the site of calcification. The coral also benefits from the oxygen produced by photosynthesis. In the confined spaces of coral tissue, oxygen levels can fluctuate. The zooxanthellae help maintain oxygen availability, particularly during the day when photosynthesis is occurring. This oxygen supports the coral's aerobic respiration, allowing it to efficiently convert sugars into usable energy. But the benefits flow both ways. The zooxanthellae receive nutrients from the coral that are scarce in the surrounding seawater. Tropical reef waters are often described as nutrient-poor, low in nitrogen and phosphorus. These are essential elements for algae, needed to build proteins and nucleic acids. The coral provides these nutrients through its metabolic waste. When the coral digests captured plankton, it produces ammonia and phosphate as byproducts. Instead of excreting these waste products into the water, the coral transfers them to the zooxanthellae. The algae use them to fuel their growth and reproduction. This recycling of nutrients is one reason coral reefs can thrive in nutrient-poor waters. The nutrients are not lost to the open ocean. They are retained within the coral-algae partnership, cycled and recycled in a tight loop. The reef becomes a nutrient island in an otherwise barren sea. The zooxanthellae also give the coral its color. The pigments within the algal cells, chlorophylls, carotenoids, and other photosynthetic pigments, create the vibrant hues that make coral reefs so visually striking. Browns, greens, yellows, and sometimes purples and reds all come from the algae. The coral polyp itself, if you were to see it without its zooxanthellae, will be nearly transparent, a pale ghost of its colorful form. Different species of zooxanthellae have different pigment compositions, which can affect the color of the coral. Some corals host multiple species of zooxanthellae simultaneously, and the dominant species can shift in response to environmental conditions. This ability to shuffle symbionts, to change which species of algae predominates within the tissue, may provide corals with some flexibility to adapt to changing environments. But that flexibility has limits. The zooxanthellae reproduce inside the coral. They divide asexually, producing daughter cells that remain within the host tissue. The coral can regulate the population of algae to some extent, digesting excess cells when populations grow too large. But the coral also needs to maintain enough zooxanthellae to meet its energy needs. If the population of algae drops too low, the coral may suffer from energy starvation. This balance is maintained through a constant exchange of signals between the coral and its symbionts. The exact nature of these signals is still being studied, but it is clear that the relationship is dynamic, not static. The coral and the algae are continuously communicating, adjusting to each other's needs. Not all corals host zooxanthellae. Some corals, particularly those living in deep water where light does not penetrate, do not form symbiotic relationships with photosynthetic algae. These corals rely entirely on capturing prey for their energy. They are called azooxanthellae corals. They grow more slowly than their symbiotic counterparts and do not build the large, reef-forming structures that characterize shallow tropical seas. The presence or absence of zooxanthellae fundamentally changes what a coral can do. With algae, a coral becomes a solar-powered organism, able to harness the energy of sunlight to build vast structures of calcium carbonate. Without algae, a coral remains dependent on the unpredictable availability of drifting food particles. The difference is the difference between a forest and a scattering of shrubs. The partnership between coral and zooxanthellae is mutualistic, meaning both partners benefit. This distinguishes it from parasitism, where one organism benefits at the expense of the other, and from commensalism, where one organism benefits and the other is neither helped nor harmed. In mutualism, both partners gain something essential. The coral gains energy and oxygen. The algae gains shelter and nutrients. Neither could achieve the same success alone. But mutualism does not mean harmony in all circumstances. The relationship can become strained. When environmental conditions change, when water temperatures rise too high, when light levels become too intense, when pollutants enter the water, the coral can become stressed. And when the coral is stressed, it may expel its zooxanthellae. This process is called coral bleaching. The coral loses its color because the colored algae are gone. What remains is the translucent polyp tissue overlaying the white calcium carbonate skeleton beneath. The coral is not dead, not yet, but it is starving. Without its zooxanthellae, it has lost its primary source of energy. If the stress passes quickly and conditions return to normal, the coral can recover. New zooxanthellae can recolonize the tissue, and the partnership can resume. But if the stress persists, if the coral remains bleached for too long, it will die. The reasons why corals expel their zooxanthellae during stress are complex and not fully understood. One hypothesis is that the algae under stress begin producing harmful reactive oxygen species, toxic molecules that damage the coral cells. The coral expels the algae to protect itself from this damage. Another hypothesis is that the coral actively breaks down the symbiosis as a way to reset the relationship, hoping to acquire more stress-tolerant algae when conditions improve. The truth may involve elements of both explanations. What is clear is that the partnership, for all its elegance and efficiency, is fragile. It has been refined over millions of years to function optimally within a narrow range of environmental conditions. When those conditions shift too far or too fast, the partnership can collapse. Yet the fact that this relationship exists at all is remarkable. 2. Organisms separated by hundreds of millions of years of evolutionary history, an animal and an alga, have evolved to live together in such close integration that their metabolic processes are intertwined. The coral provides the structure, the protection, the waste products that the algae need. The algae provide the energy, the oxygen, the fuel that powers the construction of the reef. Together, they accomplish what neither could do alone. They transform sunlight into stone. They create ecosystems that support a quarter of all marine life. They build structures visible from space. This partnership, written in light and sugar and calcium and chlorophyll, is the engine of the coral reef. It is the quiet collaboration at the heart of one of Earth's most productive ecosystems. And it is a reminder that complexity and abundance can emerge from cooperation, from the seamless integration of two simple organisms into something far greater than the sum of their parts. The reef grows because the polyp and the algae have found a way to share. The polyp shares its body. The algae share their food. And from this exchange, stone rises from the seafloor, layer by patient layer year after year for thousands of years. Time moves differently beneath the waves. While storms can reshape a coastline in hours and tides shift twice each day, the coral reef measures its existence in centuries and millennia. What appears solid and permanent is actually the cumulative result of countless small acts repeated over spans of time that exceed human comprehension. A single coral polyp, secreting calcium carbonate at the base of its body, might add 1 to 3 centimeters of skeletal material each year. This varies by species, by environmental conditions, by the abundance of light and nutrients. Some branching corals grow faster, extending their fingers upward at rates approaching 25 centimeters per year under ideal conditions. Massive boulder corals grow more slowly, adding thin layers concentrically, like tree rings, accumulating perhaps 1 centimeter of vertical growth annually. These numbers seem almost trivial. A centimeter here, a few centimeters there. But time transforms the trivial into the monumental. Consider a coral colony that grows at a rate of 2 centimeters per year. In a decade, it has grown 20 centimeters, roughly the length of a human hand. In a century, 2 meters, taller than most people. In 1,000 years, 20 meters, the height of a 6-story building. And coral reefs have been growing for far longer than 1,000 years. The Great Barrier Reef, the largest living structure on Earth, began forming in its current location approximately 6,000 to 8,000 years ago. But the story reaches back much further. The reef system has existed in various forms for hundreds of thousands of years, advancing and retreating with the ice ages. When sea levels fell during glacial periods, the reef was exposed to air and died. When the ice melted and the ocean returned, new corals settled on the foundations of the old, building upward once again. This cycle of growth, death, and regrowth has repeated many times. Each iteration leaves behind layers of limestone, compressed skeletons of corals that lived and died tens of thousands of years ago. Beneath the living veneer of modern reef lies a vast archive of calcium carbonate, meters thick in some places, kilometers in extent. The reef is not built uniformly. Different species of coral grow at different rates and in different forms. Branching corals, with their delicate tree-like structures, grow quickly but are fragile. They break easily in storms, their fragments scattered across the reef. Some of these fragments survive and continue growing where they land, starting new colonies. Others are ground into sand by wave action and the feeding activities of parrotfish and other bioeroders. Massive corals, shaped like boulders or domes, grow more slowly but are more resistant to physical damage. They can persist for centuries, weathering storms that destroy faster-growing species around them. Plate corals spread horizontally, forming flat, table-like structures that maximize their exposure to sunlight. Each growth form represents a different strategy for survival, a different way of building in the dynamic environment of the reef. The accumulated skeletons of all these corals, living and dead, form the physical structure of the reef. The living tissue, the polyps, and their zooxanthellae, occupies only the outermost surface, a thin veneer of life covering a foundation of stone. Below the surface, the reef is a complex matrix of chambers, channels, and cavities. Water flows through these spaces. Small animals burrow into them. Sponges bore tunnels. The reef is as much absence as presence, as much hollow space as solid calcium. Over time, the spaces fill with sediment, fragments of broken coral, shells of dead mollusks, the skeletal remains of coral and algae. This sediment is compacted and cemented by chemical processes, gradually transforming the loose debris into solid limestone. What was once a collection of individual skeletons becomes a unified geological formation. This process has been happening for hundreds of millions of years. Corals first appeared in the fossil record during the Ordovician period, more than 450 million years ago. The partnership with zooxanthellae appears to have begun at least 200 million years ago, during the Triassic. Since then, reef-building corals have been laying down limestone in shallow tropical seas. The limestone deposits they have created are immense. Some ancient coral reefs, now exposed on land due to geological uplift, form cliffs and mountains. The rocks of the Dolomite Mountains in Italy were once coral reefs in a warm Triassic sea. The limestone formations visible today are the skeletons of corals that lived more than 200 million years ago. Modern reefs are continuing this tradition. They are adding new layers to the geological record, creating structures that may one day be uplifted and exposed, becoming the mountains of a distant future. But for now, they remain beneath the waves, growing slowly upward toward the light. The formation of a coral reef begins with a single event, the settlement of a coral larva. Corals reproduce sexually, releasing eggs and sperm into the water column. In some species, this happens in a spectacular synchronized spawning, where entire colonies release their gametes simultaneously, turning the water milky with reproductive cells. The timing is precise, coordinated by the cycles of the moon and the temperature of the water. When sperm meets egg in the open water, fertilization occurs. The fertilized egg develops into a tiny, free-swimming larva called a planula. The planula is ciliated, covered with hair-like structures that propel it through the water. It drifts with the currents, carried by tides and waves, moving away from the parent colony. This dispersal phase can last for hours, days, or even weeks, depending on the species. During this time, the larva is vulnerable. It is food for countless predators, fish, jellyfish, and other planktonic organisms. The vast majority of coral larvae do not survive, but a few do, and those few are enough to continue the process. Eventually, if the larva survives, it begins to search for a place to settle. It needs a hard surface, a rock, a dead shell, the skeleton of another coral. It cannot settle on sand or mud. These substrates are too unstable. The larva uses chemical cues to assess potential settlement sites. It responds to the presence of coralline algae, a type of calcifying red algae that often indicates suitable habitat. It avoids surfaces covered with fleshy algae or heavy sediment. When the larva finds a suitable spot, it attaches. This is an irreversible decision. Once attached, the larva undergoes metamorphosis. It transforms from a free-swimming organism into a sessile polyp. It develops a mouth, tentacles, and a gastrovascular cavity, and it begins to secrete calcium carbonate, forming the first tiny cup of skeleton beneath its body. This single polyp is now the founder of what may become a colony spanning meters, persisting for centuries. The polyp begins to bud, producing clones of itself. The colony grows. Each new polyp adds its own calcium carbonate skeleton to the structure. The colony expands outward and upward. But the polyp does not grow in isolation. Around it, other coral larvae are settling. Some belong to the same species, others are different. Each establishes its own colony. Over time, these colonies grow and may come into contact with one another. When this happens, competition begins. Corals compete for space on the reef. Some species use their tentacles to attack neighboring colonies, stinging them with nematocysts. Others overgrow their competitors, shading them from light. The fastest-growing corals often win these battles, but they are also the most fragile, more easily broken by storms. The result is a mosaic of different coral species and growth forms, each occupying its own niche on the reef. Branching corals dominate in shallow, high-energy zones where light is abundant but wave action is strong. Massive corals thrive in areas with moderate energy. Plate corals grow in deeper, calmer waters where light is reduced and horizontal growth maximizes photosynthetic surface area. As the colonies grow and die, as storms break branches and biorotors hollow out skeletons, sediment accumulates in the spaces between living corals. This sediment, fine particles of coral skeleton, shells, sand, is cemented together by calcium carbonate precipitation, a chemical process facilitated by bacteria and other microorganisms. The reef becomes more solid, more cohesive. Over centuries, the reef structure builds upward. The growth is not continuous or uniform. There are periods of rapid expansion when conditions are favorable, when water temperatures are stable, when light is abundant, when few disturbances occur. There are periods of decline when storms break large sections of the reef, when disease sweeps through coral populations, when unusually cold or warm water kills corals. But as long as the rate of growth exceeds the rate of erosion, the reef persists and expands. And if conditions remain favorable for long enough, thousands of years, the reef can grow into one of three major structural types. Fringing reefs grow directly along coastlines attached to the shore. They are the most common type of coral reef. They form when coral larvae settle on rocky shorelines in shallow water. The reef grows outward from the coast, creating a narrow band of coral extending from the beach into the ocean. There is little or no lagoon between the reef and the shore. Fringing reefs are often found around volcanic islands, growing on the submerged slopes of the volcano. Barrier reefs also grow parallel to coastlines, but they are separated from the shore by a deep lagoon. The lagoon can be several kilometers wide and many meters deep. Barrier reefs form when fringing reefs grow upward as sea level rises, or as the island or continent they border subsides. The reef keeps pace with a changing water level, but the land behind it sinks or is eroded, creating the lagoon. The Great Barrier Reef is the most famous example, stretching over 2,300 kilometers along Australia's coast. It is not a single reef but a complex system of over 2,900 individual reefs and 900 islands. Atolls are the most distinctive reef structures. They are ring-shaped reefs surrounding a central lagoon, with no island in the middle. From the air, they appear as perfect circles or ovals of coral enclosing turquoise water. The formation of an atoll is a slow process that can take millions of years. The process begins with a volcanic island rising from the seafloor. As the volcano emerges above the water, coral larvae settle on its submerged slopes, forming a fringing reef around the island. The volcano is active initially, but eventually it becomes dormant. Once volcanic activity ceases, the island begins to erode. Rain wears away the rock. The island gradually shrinks. At the same time, the oceanic plate on which the volcano sits is moving. As the plate moves away from the volcanic hotspot that created the island, it cools and subsides, slowly sinking back toward the seafloor. The island grows lower, but the coral reef keeps growing upward, maintaining its position near the water surface. A lagoon forms between the sinking island and the reef. The fringing reef becomes a barrier reef. Eventually, the volcanic island disappears entirely beneath the waves. All that remains is the coral reef, still growing upward, still encircling the space where the island once stood. This is an atoll. This process was first described by Charles Darwin in 1842, based on his observations during the voyage of the HMS Beagle. Darwin had never seen an atoll form. The process is far too slow to observe directly, but he understood the evidence. He recognized that atolls were ancient structures, built over timescales that dwarf human history. Later investigations, including drilling into atolls to examine their internal structure, confirmed Darwin's hypothesis. Beneath the living coral surface lies hundreds of meters of dead coral skeleton, layered over the basalt core of the sunken volcano. All three types of reefs, fringing, barrier, and atoll, are built by the same process. Coral larvae settle, polyps grow and bud, colonies expand, skeletons accumulate. The difference is the geological context in which this growth occurs. A stable coastline produces a fringing reef. A subsiding coastline or rising sea level produces a barrier reef. A disappearing volcanic island produces an atoll. But regardless of type, all reefs share fundamental requirements. They need warm water, temperatures between 23 and 29 degrees Celsius. They need clear water so that sunlight can penetrate to the zooxanthellae. They need a hard substrate for larvae to settle on. They need relatively stable salinity, low nutrient levels, and minimal sediment. These requirements confine coral reefs to tropical and subtropical regions, to shallow coastal waters, and the slopes of oceanic islands. Where these conditions exist, reefs flourish. Where they do not, corals may survive as scattered individuals or small colonies, but they do not build reefs. The distribution of coral reefs across the world's oceans is a map of where the necessary conditions align. And when they do align, when larvae settle and polyps build, When colonies expand and skeletons accumulate, the result is a structure of astonishing complexity and beauty. A living architecture shaped by the slow, patient work of countless animals, repeated over thousands of years. A monument not to individual achievement, but to the power of small, sustained efforts compounded across deep time. The reef rises from the seafloor, molecule by molecule, polyp by polyp, year by year. And in this rising, this patient accumulation, it creates the conditions for something even more remarkable. It creates habitat. It creates complexity. It creates the foundation for an ecosystem that will shelter a quarter of all marine life on Earth. But first, the conditions must be right. Coral reefs do not grow everywhere. They are not scattered randomly across the ocean floor, appearing wherever currents carry coral larvae. Instead, they are confined to specific regions, specific depths, specific environments where a precise set of conditions align. The requirements are narrow. The tolerances are small. And when any single condition falls outside the acceptable range, reef growth slows or stops entirely. The first requirement is temperature. Coral reefs thrive in warm water, typically between 23 and 29 degrees Celsius. This range corresponds roughly to 73 to 84 degrees Fahrenheit. Within this window, the biochemical processes of both the coral polyp and its zooxanthellae function optimally. The coral can efficiently secrete calcium carbonate. The algae can photosynthesize at rates high enough to provide the energy the coral needs. The partnership flourishes. But temperature, like so many factors in nature, is a constraint with sharp boundaries. If the water becomes too cold, metabolic processes slow. The coral's ability to build its skeleton declines. The zooxanthellae photosynthesize less efficiently. Growth rates drop. If temperatures fall below 18 degrees Celsius for extended periods, many reef-building corals cannot survive. They stop growing. They may die. Conversely, if the water becomes too warm, the coral experiences stress. Even a temperature increase of 1 or 2 degrees above the normal summer maximum can trigger problems. The zooxanthellae begin to malfunction. Under heat stress, their photosynthetic machinery produces harmful reactive oxygen species, molecules that damage the coral cells. The coral, in response, expels the algae. This is coral bleaching, a breakdown of the essential partnership between animal and alga. Without its zooxanthellae, the coral loses its primary energy source. It can survive for a while, relying on its tentacles to capture food. Fragile in its dependence on specific environmental conditions, its sensitivity to change, its vulnerability to human impacts. When the conditions are right, life flourishes. The reef becomes a city beneath the waves, teeming with color and movement. And it is to that city, to the richness of life it supports, that the story now turns. If you could shrink yourself down and walk through a coral reef, you would find yourself in a metropolis. Not the concrete and steel cities of human construction, but a living city built of calcium carbonate and sustained by sunlight. A place where millions of organisms live, feed, reproduce, and die within a space measured in square meters. A place where every surface is occupied, every crevice inhabited, every niche filled. Coral reefs cover less than 1% of the ocean floor, yet they support an estimated 25% of all marine species. This statistic is repeated often, but its implications are staggering. In this tiny fraction of the ocean's area, life is concentrated to a degree matched only by tropical rainforests on land. More than 4,000 species of fish make their homes on coral reefs at some point in their lives. The total number of species, when you count every organism from bacteria to sharks, may exceed 1 million. Some scientists believe the true biodiversity of coral reefs is even higher, perhaps approaching 2 million species. The challenge is that many reef organisms are small, cryptic, and difficult to identify. They live in the spaces between coral branches, burrow into dead skeleton, or exist as microscopic members of the plankton community. A single square meter of reef substrate can harbor hundreds of species that have never been formally described or named by science. This density of life is not evenly distributed. The reef has structure. It has zones, each characterized by different physical conditions and dominated by different assemblages of organisms. The reef crest is the highest point of the reef, the zone that experiences the most intense wave action. Here, massive, boulder-shaped corals dominate. These corals are built to withstand the constant pounding of waves. Their rounded, compact forms shed water efficiently. They grow slowly, but are difficult to break. Among these massive corals live organisms adapted to turbulent water, sea urchins that wedge themselves into crevices, algae that cling tightly to rock surfaces, fish that can swim against strong currents. Below the crest, on the reef slope that descends toward deeper water, conditions become calmer. Wave energy dissipates. Light levels remain high, but the physical stress is reduced. Here, diversity reaches its peak. Branching corals extend their delicate structures into the water. Plate corals spread horizontally, maximizing their surface area for capturing light. The three-dimensional complexity creates countless microhabitats, overhangs, caves, channels, and platforms. Each of these microhabitats is occupied. In the crevices between coral branches, small damselfish establish territories. They defend patches of algae that grow on dead coral surfaces, farming these algae as a food source. Cleaner wrasses set up cleaning stations where larger fish come to have parasites removed. The wrasses pick parasites from the skin, gills, and even inside the mouths of their clients. This is mutualism. The wrasses get food, the larger fish get cleaned. In the sandy patches between coral formations, garden eels emerge from burrows, their bodies swaying in the current as they pluck zooplankton from the water. Rays bury themselves in the sand, camouflaged and waiting for prey. Gobies and shrimp form partnerships, the shrimp digging and maintaining a burrow while the sharp-eyed goby watches for predators, alerting its nearly blind partner with a flick of its tail when danger approaches. On the surface of dead coral skeleton, a different community thrives. Coralline algae, calcifying red algae, cement loose rubble together, helping to stabilize the reef. Structure. Sponges bore into the skeleton, creating tunnels and chambers. Bivalves and worms excavate their own hollows. These bioeroders break down the reef structure even as living corals build it up. The balance between construction and erosion determines whether the reef grows or shrinks. In the open water above the reef, schools of fish move in coordinated formations. Fusiliers swim in tight groups, thousands of individuals moving as one, creating a shimmering cloud of silver bodies. They feed on zooplankton drifting in the current. Below them, predators wait. Jacks and trevally patrol the reef edge, explosive swimmers that can accelerate rapidly to capture prey. Groupers hide in caves and under overhangs, ambush predators that strike from concealment. At night, the reef transforms. Many of the fish that were active during the day retreat into crevices and sleep. They secrete mucus cocoons around their bodies, which may help mask their scent from nocturnal predators. As the diurnal fish settle, nocturnal species emerge. Octopuses leave their dens and hunt across the reef, probing into crevices with their flexible arms. Moray eels slither out of their holes, following scent trails to locate sleeping fish. Spiny lobsters march across the seafloor in long single-file lines. The coral polyps themselves extend their tentacles at night. During the day, many species retract, relying on their zooxanthellae for energy. But at night, they become active feeders, capturing zooplankton that rises from deeper water under cover of darkness. The reef surface, smooth and colorful in daylight, becomes fuzzy with thousands of tiny tentacles waving in the current. The diversity of the reef is not just a catalog of species. It is a web of interactions, a network of relationships where every organism is connected to others through feeding, competition, reproduction, and symbiosis. Some of these relationships are predator and prey. Parrotfish, with their fused teeth forming beak-like structures, scrape algae from coral surfaces. In the process, they bite off pieces of coral skeleton, which they grind with specialized pharyngeal teeth in their throats. They excrete the ground-up calcium carbonate as fine sand. A single large parrotfish can produce hundreds of kilograms of sand each year. The white sand beaches of many tropical islands are largely composed of parrotfish excrement. Butterfly fish have small, pointed mouths adapted for reaching into coral crevices to pluck out coral polyps. Some species are specialized, feeding on only one or a few species of coral. Crown-of-thorns sea stars are voracious coral predators. They climb onto coral colonies, extend their stomachs out through their mouths, and digest the living coral tissue externally. A single crown of thorns can consume several square meters of coral in a year. When these sea stars undergo population explosions, they can devastate large areas of reef. Other relationships involve competition for space. Corals grow slowly, but they are long-lived, and over time they can spread across large areas. When two coral colonies of different species meet, they compete. Some corals extend specialized sweeper tentacles that are longer and more heavily armed with nematocysts than their feeding tentacles. These sweeper tentacles can reach out and sting neighboring corals, killing their tissue and creating space for expansion. Other corals produce chemicals that inhibit the growth of competitors. Some simply overgrow their neighbors, shading them from light. Algae and coral compete as well. Fleshy macroalgae can overgrow coral colonies if herbivore populations are low. The algae shade the coral, blocking light from reaching the zooxanthellae. They can also release chemicals that stress the coral or interfere with coral larval settlement. Maintaining the balance between coral and algae depends on herbivores, parrotfish, surgeonfish, sea urchins, that graze the algae and prevent it from overgrowing the reef. Symbiotic relationships extend far beyond the coral zooxanthellae partnership. Many species of snapping shrimp live in association with gobies. The shrimp excavates a burrow in the sand or rubble, providing shelter for both partners. The goby, with better eyesight, stands guard at the burrow entrance. When a predator approaches, the goby signals the shrimp by touching it with its tail, and both retreat into the burrow. The shrimp provide housing. The goby provides security. Anemonefish live among the tentacles of sea anemones. The anemone's tentacles are armed with nematocysts that sting and kill most fish that touch them. But anemonefish have a specialized mucus coating that protects them from the stings. They gain protection from predators by hiding among the anemone's tentacles. In return, the anemonefish may defend the anemone from butterfly fish that prey on anemone tentacles, and their movements may help circulate water around the anemone. Cleaner organisms, wrasses, shrimp, and even some juvenile fish provide a service by removing parasites and dead tissue from larger fish. They establish cleaning stations, recognizable locations on the reef where client fish come to be cleaned. The cleaners perform elaborate dances to advertise their services. Large predatory fish that would normally eat small fish will allow cleaners to enter their mouths and gills, remaining still while being cleaned. The cleaners gain food. The clients gain health. The reef also provides nursery habitat for many fish species that spend their adult lives elsewhere. Mangrove forests and seagrass beds near reefs serve as nurseries for juvenile fish, but the reef itself shelters the young of countless species. The complex structure provides hiding places from predators. The abundance of small invertebrates provides food. As the fish grow, they move to different parts of the reef or into deeper water. Reproduction on the reef follows many strategies. Some fish form monogamous pairs that defend territories and care for their eggs. Clownfish, damselfish, and jawfish are examples. The parents guard the eggs, fanning them with their fins to keep them oxygenated and removing any that become infected with fungus. Other fish gather in massive spawning aggregations. Groupers, snappers, and surgeonfish may travel hundreds of kilometers to specific spawning sites. Hundreds or thousands of individuals gather at the same location, often synchronized with the lunar cycle. They release eggs and sperm simultaneously into the water. The fertilized eggs drift with the currents, and the larvae that hatch may be carried far from their birthplace before settling back onto a reef. Corals themselves engage in spectacular synchronized spawning events. On certain nights of the year, often a few days after the full moon, entire reefs of coral release eggs and sperm simultaneously. The water becomes thick with gametes, a milky cloud that can cover kilometers of reef. The synchronization increases the chances of fertilization and may overwhelm predators with the sheer abundance of food. Timing is controlled by environmental cues, water temperature, day length, and lunar cycles. The reef is not isolated from the surrounding ocean. Nutrients and larvae flow onto the reef with currents. Fish that feed on the reef may travel into open water or to adjacent habitats. Seabirds nest on reef islands and deposit guano that enriches the waters around a reef. Whales and dolphins pass by, sometimes interacting with reef organisms. The reef is a node in a larger network of ocean ecosystems. The abundance of life on the reef exists within a paradox. The surrounding waters are nutrient-poor, often described as oceanic deserts. Yet the reef teems with productivity. How can so much life thrive in such apparently barren seas? The answer lies in efficiency and recycling. On the reef, nutrients are not lost. They are captured, used, recycled, and used again. The tight coupling between coral and zooxanthellae is part of this. The coral's waste products feed the algae. The algae's photosynthetic products feed the coral. Nutrients cycle within the partnership rather than being released into the water. The same principle applies at the ecosystem level. When a fish dies, its body does not drift away into the open ocean. It is consumed by scavengers, crabs, worms, bacteria. The nutrients in its tissues are broken down and reincorporated into the reef community. Fecal matter from fish and invertebrates is consumed by detritivores or decomposed by bacteria, returning nutrients to the system. The physical structure of the reef helps retain nutrients. The complex three-dimensional architecture creates areas of reduced water flow where particles settle. Mucus produced by corals and other organisms traps fine particles, which are then consumed by filter feeders. Little is wasted. This efficiency allows the reef to achieve high productivity despite low nutrient inputs from the surrounding water. The reef ecosystem is essentially oligotrophic, adapted to low nutrient conditions. It functions through tight nutrient recycling rather than through high nutrient availability. But this efficiency comes with fragility. Because the system is finely tuned and tightly coupled, disruptions can cascade through the community. If herbivore populations decline, algae can overgrow corals. If coral cover decreases, fish populations that depend on coral for shelter or food decline. If key predators are removed, prey populations may explode and overconsume their own resources. The reef is a community held together by countless invisible threads: predation, competition, symbiosis, and facilitation. Remove one thread and the fabric may remain intact. Remove many threads and the structure can unravel. The richness of the reef, the sheer number of species, the complexity of their interactions, the density of life packed into small spaces, is both its strength and its vulnerability. Diversity can provide resilience. If one species declines, another may fulfill a similar ecological role. But diversity also means that there are many species with specialized requirements, narrow niches, and limited distributions. These specialists are the first to disappear when conditions change. Standing on a reef, or floating above one with a mask and snorkel, you see color and movement. Fish darting between coral branches. Sea turtles gliding past. Rays buried in the sand with only their eyes protruding. But for every organism you see, there are hundreds or thousands you do not. The tiny invertebrates living between coral polyps. The bacteria are colonizing every surface. The larvae drifted in a water column, invisible to the naked eye. The reef is a city, but not the city you see. It is the city beneath the surface. The city in the crevices. The city of chemical signals, of partnerships formed over millions of years of evolution, of energy flowing from sunlight to algae to coral to fish to predators and back to the reef when organisms die. It is a city built of calcium carbonate but animated by carbon from photosynthesis. A city where the architecture creates the conditions for habitation and the inhabitants, in turn, shape the architecture. A city where life is concentrated because the conditions allowed it, and where that concentration of life has created opportunities for even more life. The reef as a city is both metaphor and reality. It is a dense aggregation of organisms living in close proximity, their lives intertwined. It is a place of constant activity, constant change, constant creation and destruction. It is a place where energy and materials flow through networks of consumption and production. And like any city, it depends on infrastructure. The coral skeleton is the infrastructure of the reef. It provides the foundation, the surfaces, the shelter that makes everything else possible. Without the coral's patient work of calcification, there would be no reef. Without the reef structure, there would be no habitat for the thousands of species that make the reef their home. The energy that powers the city flows in from outside. Sunlight, captured by zooxanthellae and by algae growing on dead coral surfaces, enters the system as chemical energy. Plankton, drifting in from the open ocean, brings additional energy and nutrients. And from these inputs, the reef community builds itself, millions of organisms, each playing a role, each connected to others through the flow of energy and the exchange of resources. This flow of energy, this movement of resources from organism to organism, is the circulation system of the reef city. And it is to this flow, to the pathways through which energy moves across the reef, that the story now turns. Energy does not appear from nowhere. It flows. It moves from one organism to another, transformed with each transfer, diminished with each step, yet essential to every living thing on the reef. Understanding the reef means understanding this flow, where energy enters the system, how it moves through the community, and where it finally dissipates. The story of energy on a coral reef begins, as most stories of life do, with the sun. Sunlight penetrates the clear tropical water, reaching the shallow depths where coral reefs grow. This light carries energy, electromagnetic radiation that has traveled 150 million kilometers from the sun's core to reach the reef. Most of this energy is reflected or absorbed by the water, converted to heat. But some is captured. The primary captors of solar energy on a reef are photosynthetic organisms. These are the producers, the organisms that convert light energy into chemical energy through photosynthesis. They form the base of the reef food web. The most important producers are the zooxanthellae living inside coral tissues. These microscopic algae, numbering in the billions across the reef, photosynthesize continuously during daylight hours. They produce sugars, glucose, glycerol, amino acids that fuel not only their own metabolism but also that of their coral hosts. Up to 90% of the organic compounds produced by zooxanthellae are transferred directly to the coral polyp. This energy powers coral growth, reproduction, and the essential process of calcification that builds the reef structure. But zooxanthellae are not the only producers. Tiny single-celled phytoplankton drift in the water column above the reef. They are so small that hundreds could fit on the head of a pin. Yet collectively, they capture significant amounts of solar energy. These phytoplankton are the free-floating cousins of zooxanthellae, independent organisms rather than symbionts. On the surfaces of dead coral skeleton and on bare rock, other algae grow. Turf algae form low, carpet-like mats. Coralline algae deposit calcium carbonate as they grow, cementing loose rubble and helping stabilize the reef structure. Macroalgae, larger, fleshy seaweeds, colonize areas where coral cover has declined. All of these organisms photosynthesize. All capture light and convert it to chemical energy. Together, these producers form the first trophic level of the reef food web. A trophic level is simply a position in the sequence of energy transfer. Producers occupy the first level because they create energy-rich organic compounds from sunlight, water, and carbon dioxide. They do not need to consume other organisms to obtain energy. They harvest it directly from the sun. The energy captured by producers becomes available to other organisms through consumption. This is where the second trophic level begins, the herbivores, organisms that eat plants and algae. Herbivores on the reef are diverse. Parrotfish, with their beak-like teeth, scrape algae from coral surfaces. In the process, they also bite off and ingest small amounts of coral skeleton, which they grind with pharyngeal teeth in their throats before excreting as fine sand. A large parrotfish can consume and process several kilograms of reef material each day, extracting the algae and organic matter coating the surface. Surgeonfish, named for the sharp blade-like spines at the base of their tails, are also important herbivores. They graze on turf algae and macroalgae, moving across the reef in small groups. Their constant grazing helps prevent fleshy algae from overgrowing corals. Sea urchins feed on algae as well. They move slowly across the reef, using their 5-part mouths to scrape algae from rock surfaces. In some reef systems, sea urchins are the dominant herbivores, controlling algal growth and maintaining the balance between coral and algae. Even some fish that might seem too small to matter play important herbivorous roles. Damselfish defend territories on the reef, cultivating patches of algae that they farm as a food source. They aggressively chase away other herbivores, allowing dense algal mats to grow within their territories. Larger herbivores exist as well. Sea turtles, particularly green sea turtles, graze on seagrasses and algae in reef-adjacent habitats. While they may not feed directly on the reef itself, they are part of the broader ecosystem and rely on the productivity of coral reef environments. When a parrotfish consumes algae, energy is transferred from producer to herbivore. But this transfer is inefficient. Much of the energy contained in the algae is lost. Some is used by the algae's own metabolism before being eaten. Some is not digested by the parrotfish and is excreted. Some is used by the parrotfish for its own metabolic processes, swimming, maintaining body temperature, cellular respiration. Only about 10% of the energy contained in the algae ends up stored in the parrotfish's tissues as growth. This inefficiency is a fundamental characteristic of energy transfer in ecosystems. Energy cannot be created or destroyed, but it can be transformed. And with each transformation, some energy is lost as heat. This is why food webs are often depicted as pyramids, with a broad base of producers supporting a smaller mass of herbivores. But the herbivores are not the end of the chain. They, in turn, become food for predators. The third trophic level consists of carnivores, organisms that feed on herbivores. These are the predators of the reef, and they are varied in their hunting strategies. Some predators are ambush hunters. Groupers hide in caves or beneath overhangs, their mottled coloration blending with the shadows. They remain motionless, sometimes for hours, waiting for an unwary fish to swim too close. When prey approaches, the grouper strikes with explosive speed, opening its large mouth and creating a powerful suction that draws the prey inside. Moray eels are also ambush predators. They den in crevices in the reef, their long bodies coiled within the rock. Only their heads protrude, watching. When a fish or octopus passes near, the eel lunges forward, its jaws lined with sharp, backward curved teeth that prevent prey from escaping. Other predators are active hunters. Jacks and trevally patrol the reef in schools, swimming constantly, scanning for prey. When they spot a school of smaller fish, they attack with coordinated speed, driving the prey against the reef or toward the surface where escape is limited. Their streamlined bodies are built for sustained swimming and quick acceleration. Barracuda are solitary hunters, long and torpedo-shaped, with mouths filled with razor-sharp teeth. They hover motionless in the water column, then accelerate suddenly to intercept passing fish. Their hunting style relies on surprise and speed. Some carnivores specialize in particular types of prey. Butterfly fish use their elongated, pointed snouts to pluck coral polyps from their skeletons. Some species are highly specialized, feeding on only one or a few coral species. Others feed on small invertebrates hiding in coral crevices. Wrasses pick parasites from the skin and gills of larger fish. Octopuses hunt at night, using their flexible arms to probe into crevices for crabs, shrimp, and mollusks. Each time a predator consumes a herbivore, energy is transferred upward through the food web. A grouper eating a parrotfish receives some of the energy that the parrotfish obtained from eating algae, which in turn obtained energy from sunlight. But again, the transfer is inefficient. Much of the parrotfish's body is not digestible: bones, scales, indigestible proteins. Much of the energy is used by the grouper's metabolism. Only about 10% ends up as growth in the grouper's tissues. This means that the energy originally captured by the algae has been reduced to about 1% by the time it reaches the grouper. 10% from algae to parrotfish, then 10% of that from parrotfish to grouper. The pyramid narrows. But the food web does not end with these mid-level carnivores. There are higher predators still, the apex predators that feed on other carnivores. Sharks patrol the deeper waters around a reef. Reef sharks, blacktip, whitetip, gray reef sharks, hunt fish, octopuses, and crustaceans. They are the top predators in many reef systems. Their presence influences the behavior of prey species throughout the food web. Fish near the reef edge are more cautious, more vigilant, because sharks are present. Large groupers, once they reach significant size, may shift from being mid-level predators to apex predators. A grouper weighing 40 or 50 kilograms can consume other carnivorous fish, squid, even small sharks. It occupies the fourth or even fifth trophic level, far removed from the producers at the base. Barracuda, too, can function as apex predators, particularly when hunting in open water above the reef. They feed on jacks, snappers, and other mid-sized carnivorous fish. With each step up the food chain, energy is lost. By the time energy reaches an apex predator like a large shark, only a tiny fraction of the original solar energy captured by algae remains. This is why apex predators are relatively rare. The ecosystem simply cannot support large numbers of top predators. There is not enough energy available. The linear description of the food chain, algae to parrotfish to grouper to shark, is a simplification. In reality, the flow of energy on a reef is more accurately described as a food web, not a chain. Most organisms do not feed on just one type of prey. Parrotfish eat multiple species of algae. Groupers eat parrotfish but also eat snappers, wrasses, and crustaceans. Sharks eat groupers but also eat smaller fish that might be herbivores or lower-level carnivores. Energy flows through multiple pathways simultaneously. Some energy moves from algae to sea urchins to triggerfish to sharks. Other energy moves from phytoplankton to zooplankton to planktivorous fish to barracuda. Still other energy flows from zooxanthellae to coral polyps to crown-of-thorns sea stars to triggerfish. The web is intricate, with many threads connecting producers to top predators through various intermediaries. There are also organisms that do not fit neatly into the producer-herbivore-carnivore sequence. These are the detritivores and decomposers, organisms that feed on dead organic matter. When a fish dies, its body does not disappear. It sinks to the reef and is quickly discovered by scavengers. Crabs tear at the flesh. Bristleworms consume soft tissues. Bacteria colonize the carcass, breaking down complex organic molecules into simpler compounds. This decomposition releases nutrients back into the reef system, making them available to producers. Fecal matter from fish and invertebrates is also consumed by detritivores or broken down by bacteria. Mucus produced by corals, sometimes released in thick strands, captures fine particles from the water. This mucus, rich in organic matter, is eaten by various invertebrates and fish. Even the tiny fragments of coral skeleton ground up by parrotfish contain organic matter that is consumed by detritivores in the sand. This pathway from dead organisms and waste products back to producers completes the nutrient cycle on the reef. Energy cannot be recycled. It flows in one direction, from the sun through the food web, and is eventually lost as heat. But nutrients can be recycled. Nitrogen, phosphorus, and other essential elements are released when organisms die or excrete waste, and these nutrients are taken up by producers to fuel new growth. The efficiency of this recycling is one reason coral reefs can be so productive despite growing in nutrient-poor waters. Very little is wasted. Nutrients are captured, used, released, and captured again. The reef retains its nutrients rather than losing them to the surrounding ocean. The coral polyp itself occupies a unique position in the food web. It is both a producer through its symbiotic zooxanthellae and a consumer through its tentacles that capture zooplankton. Most of its energy comes from photosynthesis, but it supplements this with heterotrophic feeding. At night, when many species of zooplankton rise from deeper water into the shallows, coral polyps extend their tentacles and capture these drifting organisms. The energy captured through heterotrophic feeding is small compared to the energy provided by zooxanthellae, but it is not insignificant. It provides nitrogen and phosphorus that the algae need but cannot obtain easily from seawater. It provides amino acids for building proteins. And in corals that live in deeper water or in shaded areas where light is limited, heterotrophic feeding becomes more important, sometimes providing the majority of the coral's energy. The coral, then, is a bridge between different energy pathways. It captures energy directly from sunlight through its algae. It captures energy from the planktonic food web through its tentacles. And it provides energy to coral predators, butterfly fish, crown-of-thorns sea stars, some species of snails and worms that consume coral tissue. The calcium carbonate skeleton, once the polyp dies, becomes substrate for algae, which are then consumed by herbivores. Even in death, the coral continues to play a role in the flow of energy across the reef. The food web of a coral reef is not static. It changes with the seasons, with the time of day, with disturbances like storms or disease outbreaks. During the day, planktivorous fish swim in the water column, capturing zooplankton. At night, these fish hide, and different species, nocturnal predators, emerge to hunt. The flow of energy shifts as the cast of characters changes. When a disturbance occurs, a storm that breaks coral, a bleaching event that kills coral, an outbreak of crown-of-thorns sea stars, the food web responds. If coral cover declines, the algae that rely on dead coral surfaces may increase. This can support larger populations of herbivores. But if coral continues to decline and is replaced by fleshy macroalgae, the herbivores may not be able to keep pace with algal growth. The reef can shift from a coral-dominated to an algae-dominated state, with cascading effects throughout the food web. Conversely, if herbivore populations decline, due to overfishing, disease, or other factors, algae can overgrow corals even without other disturbances. The balance tips. Energy still flows through the system, but the pathways change. An algae-dominated reef may support different fish communities, with fewer coral-associated species and more herbivores. The flow of energy through the reef is ultimately limited by the amount of light that reaches the producers. Coral reefs exist in shallow water because that is where light penetrates. Below about 150 meters, there is not enough light to support the high rates of photosynthesis needed to fuel a diverse food web. The reef's productivity is constrained by the amount of solar energy available. But within this constraint, the reef has become remarkably efficient. Energy is captured by multiple types of producers: zooxanthellae, phytoplankton, turf algae, macroalgae, coralline algae. It flows through multiple pathways to herbivores, omnivores, and carnivores at various trophic levels. It is retained in the system through efficient nutrient recycling, and it supports a community of organisms so diverse that scientists are still discovering new species on reefs that have been studied for decades. The flow of energy is invisible. You cannot see it moving from algae to fish to shark. But it is always happening. Every bite, every capture of prey, every morsel consumed represents a transfer of energy. The reef exists because this energy flows. The colors, the movement, the abundance of life, all are manifestations of solar energy captured, transformed, and passed from organism to organism. And at the foundation of it all, at the beginning of the flow, are the microscopic cells living inside coral tissues. The zooxanthellae, photosynthesizing, converting sunlight into sugar, fueling the partnership that builds the reef. Without them, the flow would be reduced to a trickle. With them, it becomes a flood that sustains a quarter of all marine life on Earth. The flow of energy powers the reef. But the reef itself is built by the coral polyp, layer by layer, molecule by molecule. And to understand how this building happens, to see the coral not as a colony, but as an individual structure, requires looking more closely at the anatomy of the polyp itself. To understand how coral reefs are built, you must first understand the builder. Not the colony, not the reef, but the individual polyp, a single animal smaller than your fingernail. This is where the story becomes intimate, where complexity collapses into simplicity, where you see that one of Earth's most productive ecosystems is constructed by creatures of remarkable anatomical economy. A coral polyp is, at its most basic, a tube. Imagine a hollow cylinder with walls made of 2 layers of tissue, separated by a thin gel-like substance. One end of this cylinder is closed. This is the base attached firmly to the skeleton beneath. The other end is open. This is the top where the mouth resides. The mouth is not like yours. It is a single opening that serves every digestive function. Food enters through it. Waste exits through it. There is no separate anus, no one-way digestive tract. The mouth opens into the gastrovascular cavity, the central chamber of the polyp where all digestion occurs. Surrounding the mouth, arranged in a radial pattern, are the tentacles. The number varies by species. Some corals have 6 tentacles, others have 8, still others have multiples of 6 extending in concentric rings. These tentacles are hollow extensions of the body wall. They can contract and extend. During the day, many coral species retract their tentacles completely, pulling them inward so that the polyp appears as little more than a bump on the coral surface. But at night, the tentacles extend. They reach outward into the water column, fingers searching the current. The tentacles are covered with specialized cells called cnidocytes. These cells give the phylum Cnidaria its name. Each cnidocyte contains a structure called nematocyst, a coiled, thread-like tubule housed inside a capsule. The nematocyst is under tremendous internal pressure. At the surface of the cnidocyte is a trigger, a tiny hair-like projection called a cnidocyl. When prey, copepod, a tiny fish larva, a fragment of zooplankton, brushes against the cnidocyl, the nematocyst fires. The discharge happens in less than a millisecond, making it one of the fastest cellular processes in biology. The coiled thread explodes outward, turning inside out as it shoots from the capsule. The thread is barbed, armed with backward-facing spines. It pierces the prey's body and injects venom. Different types of nematocysts serve different functions. Some are designed to pierce and inject toxins. Others are sticky, adhering to the prey's surface. Still others coil around appendages, entangling the prey. A single tentacle may contain thousands of nematocysts, each ready to fire independently. The venom varies in potency depending on the coral species. Some corals produce toxins strong enough to deter fish and even cause painful stings to humans. Others produce milder venoms effective primarily against small invertebrates. But the purpose is always the same: to immobilize prey. Once the prey is stung and paralyzed, the tentacles contract, drawing it toward the mouth. The mouth opens. It can expand considerably to accommodate prey nearly as large as the polyp itself. The prey is pulled inside, into the gastrovascular cavity. The gastrovascular cavity is the polyp's stomach, intestine, and circulatory system combined into one chamber. It is lined with tissue called the gastrodermis, the inner layer of the polyp's body wall. Specialized cells in the gastrodermis secrete digestive enzymes into the cavity. These enzymes begin breaking down the prey's tissues, proteins, fats, carbohydrates. Digestion in a coral polyp is partly extracellular and partly intracellular. The enzymes secreted into the gastrovascular cavity break down the prey into smaller fragments. These fragments are then engulfed by cells lining the cavity through a process called phagocytosis. Once inside these cells, digestion continues. Nutrients are absorbed and distributed throughout the polyp's tissues. What cannot be digested, skeletal fragments, chitin, other indigestible materials, is expelled back out through the mouth. The polyp contracts its body, forcing the waste material out into the water. The mouth closes, and the polyp returns to its resting state, tentacles extended, waiting for the next meal. The efficiency of this simple system is remarkable. The polyp has no brain to coordinate these actions. It has no centralized nervous system. Instead, it possesses a nerve net, a diffused network of neurons that spreads throughout the body wall. When the tentacles detect prey, signals travel through this nerve net, triggering the coordinated response of tentacle contraction and mouth opening. It is simple reflex, but it is effective. The polyp also has no specialized respiratory or circulatory organs. Oxygen dissolved in seawater diffuses directly through the polyp's tissues. The thinness of the body wall, only 2 cell layers thick in most places, means that no cell is far from the surrounding water. Carbon dioxide, the waste product of respiration, diffuses out in the same way. The gastrovascular cavity itself aids in circulation. Water flows in and out of the cavity through the mouth, carrying oxygen and nutrients to internal tissues and removing waste products. The beating of cilia lining the cavity helps maintain this flow. The body wall of the coral polyp consists of 3 distinct layers. Understanding these layers is essential to understanding how the polyp functions, how it partners with zooxanthellae, and how it builds its skeleton. The outermost layer is the epidermis. This is the polyp's skin, the tissue in direct contact with seawater. The epidermis is composed of several cell types, each serving specific functions. Epithelial cells form a continuous covering, protecting the polyp from the external environment. Mucus-secreting cells produce a coating of mucus that covers the polyp's surface. This mucus serves multiple purposes. It traps sediment particles that settle on the coral, preventing them from smothering the polyp. The mucus can be shed, carrying away accumulated sediment. It also contains antimicrobial compounds that protect the polyp from bacterial and fungal infections. The mucus is sticky and can trap fine organic particles from the water, which the polyp then consumes, a form of filter feeding that supplements the energy obtained from zooxanthellae and captured prey. Also embedded in the epidermis are the cnidocytes containing nematocysts. These are particularly concentrated on the tentacles, but are present across the entire outer surface of the polyp. Some corals have specialized sweeper tentacles, longer tentacles with higher concentrations of nematocysts, used not for feeding, but for defense and competition with neighboring corals. Beneath the epidermis lies a thin, gelatinous layer called the mesoglia. The mesoglia is acellular, composed primarily of a protein matrix similar to collagen. It is thin, often just a few micrometers thick. Despite its thinness, the mesoglia provides structural support. It acts as a flexible skeleton, maintaining the polyp's shape while allowing the tentacles and body to contract and extend. The mesoglia is not metabolically active. It contains no cells, no blood vessels, no nerves. It is simply a physical layer separating the 2 active tissue layers. But its presence is crucial. It provides the polyp with enough structural integrity to maintain form without the rigidity that would prevent movement. The innermost layer is the gastrodermis. This is the tissue lining the gastrovascular cavity. Like the epidermis, the gastrodermis consists of several cell types. Gland cells secrete digestive enzymes into the cavity. Nutrient-absorbing cells take up digested food molecules. Ciliated cells create water currents that circulate water through the cavity, facilitating gas exchange and waste removal. But the most important feature of the gastrodermis, the characteristic that defines reef-building corals and separates them from their non-symbiotic relatives, is the presence of zooxanthellae. These microscopic algae reside within the cells of the gastrodermis. They are not free-floating in the gastrovascular cavity. Each algal cell is housed within a gastrodermal cell, enclosed in a membrane-bound compartment called a symbiosome. The symbiosome keeps the algae separate from the host cell's internal machinery, while still allowing the exchange of materials across the membrane. Nutrients produced by the algae, sugars, amino acids, glycerol, pass through the symbiosome membrane into the coral cell. Waste products from the coral's metabolism, ammonia, carbon dioxide, pass in the opposite direction into the symbiosome where the algae can use them. The density of zooxanthellae in the gastrodermis is extraordinary. A single square centimeter of coral tissue can contain more than 1 million algal cells. In a large coral colony, the total population of zooxanthellae can number in the trillions. These algae give the coral its color. The browns, greens, and yellows characteristic of healthy coral come from the photosynthetic pigments, chlorophylls and carotenoids, within the algal cells. The translucency of the polyp's tissues is not accidental. Light must penetrate through the epidermis and mesoglea to reach the zooxanthellae in the gastrodermis. If the tissues were opaque, photosynthesis would be impossible. The entire architecture of the polyp is designed to facilitate this partnership. The polyp also possesses sensory capabilities. Though it has no eyes, no ears, no nose in the conventional sense, it can detect changes in its environment. Photoreceptor cells allow the polyp to sense light levels, helping to regulate tentacle extension and other behaviors synchronized with day-night cycles. Chemoreceptor cells detect dissolved chemicals in the water, allowing the polyp to sense prey, respond to threats, and recognize chemical signals from neighboring polyps in the colony. When stressed by high temperature, intense light, pollutants, or physical damage, the polyp can respond. It may increase mucus production. It may contract its tissues. In extreme stress, it may expel its zooxanthellae, the process known as bleaching. This response is controlled by chemical signals, reactive oxygen species, stress hormones that travel through the polyp's tissues. The polyp is also capable of limited regeneration. If a portion of the polyp is damaged, torn by a predator, broken by a wave, the remaining tissue can repair itself. New cells divide and differentiate, replacing lost tissue. Small injuries can heal completely within days or weeks. Larger injuries may leave permanent scars, areas where the skeleton is exposed because the overlying tissue never fully regenerates. The coral polyp, despite its simplicity, is perfectly adapted to its dual role as both predator and farmer. By night, it captures prey with its tentacles, obtaining nitrogen, phosphorus, and other nutrients that are scarce in reef waters. By day, it relies on its zooxanthellae to provide the bulk of its energy needs through photosynthesis. This combination allows the polyp to thrive in environments where neither strategy alone would be sufficient. The size of coral polyps varies considerably among species. Some polyps are less than 1 millimeter in diameter, barely visible to the naked eye. These small polyp stony corals, such as species in the genus Acropora and Poecilopora, form delicate branching structures. Other species have polyps several centimeters across. Large polyp stony corals, such as brain corals and some mushroom corals, have polyps that are clearly visible and individually distinct. But regardless of size, all reef-building coral polyps share the same basic anatomy. A mouth surrounded by tentacles, a gastrovascular cavity for digestion, a body wall composed of epidermis, mesoglea, and gastrodermis. And in the gastrodermis, billions of zooxanthellae. This simple body plan has remained essentially unchanged for hundreds of millions of years. Corals belonging to the order Scleractinia, the stony corals that build modern reefs, first appeared in the fossil record during the Triassic period more than 200 million years ago. Their basic anatomy is the same today as it was then. The polyp's design has proven so effective that evolution has not needed to modify it substantially. What has changed? What has allowed corals to build the massive structures we recognize as reefs is what happens beneath the polyp. At the base of the polyp, where the epidermis contacts the hard substrate, specialized cells secrete calcium carbonate. This process, called calcification, occurs in a thin layer of tissue between the polyp and its skeleton. The cells actively transport calcium and carbonate ions from seawater and deposit them as crystalline aragonite. The polyp does not build its skeleton once and then stop. Calcification is a continuous process. As long as the polyp is alive, it continues to add calcium carbonate beneath itself. The skeleton grows thicker, more robust. The polyp sits atop this growing foundation, a living cap on a structure of stone. This is how the reef is built. Not by any single grand construction project, but by millions of polyps, each adding calcium carbonate molecule by molecule, day after day, year after year. The polyp is the worker. The skeleton is the monument. And the reef is the cumulative result of this labor, sustained across millennia. The polyp, in its anatomical simplicity, embodies an elegant solution to the challenge of survival in nutrient-poor tropical seas. It is small enough that diffusion alone can meet its gas exchange needs. It is simple enough that a diffused nerve net can coordinate its behaviors. Yet it is complex enough to house a thriving population of symbiotic algae, to capture and digest prey, to produce mucus and toxins, to sense its environment and respond to threats. The polyp is both ancient and modern. Its body plan dates back hundreds of millions of years. Yet it remains perfectly suited to the conditions of the modern reef. It is fragile, soft tissue, vulnerable to predators, disease, and environmental stress. But it is also resilient, capable of healing from injury, of expelling and reacquiring symbionts, of reproducing both sexually and asexually to propagate its genes. When you look at a coral reef, you see color and structure, fish and movement. But beneath it all, at the foundation of everything, is the polyp. A tube of tissue, no larger than a grain of rice, engaged in the patient work of building, capturing prey, housing algae, secreting stone, simple in form but profound in consequence. The anatomy of the polyp explains how coral reefs are possible. But to understand the enduring nature of reefs, to see why they persist for thousands of years, you must look not at the living tissue but at what lies beneath. At the skeleton, the calcium carbonate structure that the polyp builds and that remains long after the polyp itself has died. It is this skeleton, accumulating layer upon layer, that transforms the work of individual polyps into geological formations. Beneath the living tissue of every coral polyp lies a structure that will outlast the polyp by thousands of years. It is not flesh. It is not alive. Yet it is the most enduring part of the coral, the foundation upon which the entire reef is built. This is the skeleton, a precisely engineered architecture of calcium carbonate that the polyp secretes beneath itself, molecule by molecule, throughout its life. The skeleton serves multiple purposes. It provides structural support, allowing the soft-bodied polyp to maintain its shape against the constant motion of water. It offers protection from predators, a hard barrier beneath the vulnerable tissue. It anchors the polyp to the substrate, preventing it from being swept away by currents. And most importantly, it provides the permanent foundation that allows coral colonies to grow upward and outward, building structures that persist long after individual polyps have died. The skeleton begins to form as soon as a coral larva settles and metamorphoses into a polyp. Within hours of attachment, the newly transformed polyp begins secreting calcium carbonate at its base. This initial secretion forms a thin basal plate, a flat foundation that cements the polyp to the hard substrate beneath. The basal plate is the polyp's anchor, its permanent connection to the reef. As the polyp continues to secrete calcium carbonate, walls begin to rise around its base. These walls, called the theca, form a cup-shaped structure that surrounds the lower portion of the polyp's body. The theca provides lateral support, protecting the polyp's sides while leaving the top, the oral disc with its mouth and tentacles, exposed to the water. Within the theca, the polyp secretes additional structures called septa. These are thin, vertical blades of calcium carbonate that radiate inward from the theca walls toward the center of the cup. The septa provide additional structural support and increase the surface area for calcification. They also create channels through which the polyp's tissues extend, connecting the polyp more firmly to its skeleton. The cup formed by the basal plate, theca, and septa is called a corallite. Each polyp sits within its own corallite, custom-built chamber that fits the polyp's body precisely. The living tissue extends down into the corallite, adhering to the calcium carbonate walls. The polyp and its skeleton are intimately connected, the soft tissue draped over the hard structure like a glove. But the skeleton is not built once and then abandoned. Calcification continues throughout the polyp's life. The polyp secretes new layers of calcium carbonate beneath itself constantly, adding to the thickness of the basal plate and the height of the theca walls. Over time, the corallite grows deeper and more robust. Old layers of skeleton are buried beneath new ones. The process of calcification occurs in a thin layer of tissue between the polyp's base and the skeleton. Here, specialized cells actively transport calcium and carbonate ions from seawater. These ions are concentrated in the calcification space, where they combine to form calcium carbonate crystals. The crystals that form are typically aragonite, a specific crystalline structure of calcium carbonate that is relatively dense and strong. The formation of aragonite from dissolved ions requires energy. The coral must actively pump calcium ions to the calcification site. It must maintain the chemical conditions, the pH and ion concentrations, that favor crystal formation. This is where the partnership with zooxanthellae becomes critical. The energy provided by algal photosynthesis powers the calcification process. Without this energy subsidy, the coral could not build its skeleton at the rates necessary to construct reefs. In colonial corals, the species that form most reef structures, individual polyps do not grow in isolation. As a polyp buds, producing genetically identical offspring, the new polyp forms its own corallite adjacent to the parents. The walls of neighboring corallites may fuse together, creating a continuous skeletal structure. Between and around the individual corallites, the colony secretes additional calcium carbonate called choanostium. This is the shared skeleton that unites the colony. Above the choanostium lies the choanenchyme, the living tissue that connects individual polyps. Through this tissue run gastrovascular canals, tubes that link the polyps' internal cavities. Nutrients, chemical signals, and cellular materials flow through these canals, allowing the colony to function as an integrated unit rather than a collection of individuals. The architecture of the skeleton varies dramatically among coral species. Branching corals, such as species in the genus Acropora, build delicate, tree-like structures with corallites arranged at the tips of slender branches. These skeletons are light and porous, maximizing surface area for polyps and zooxanthellae while minimizing the amount of calcium carbonate needed. But this design makes them fragile, easily broken by storms or predators. Massive corals build boulder-shaped skeletons with thick walls and densely packed corallites. These structures are heavy and solid, able to withstand strong wave action. They grow slowly, adding just a few millimeters of skeleton each year, but they cannot persist for centuries. Some massive coral colonies are over 1,000 years old, their skeletons containing layers that span human history. Plate corals form horizontal, table-like structures. Their skeletons are flat on top, with corallites arranged across the upper surface. This design maximizes the area exposed to sunlight, allowing the coral to capture light efficiently in deeper water where light levels are reduced. The underside of the plate is supported by thin pillars of calcium carbonate, creating an architecture that is both light and strong. The coral skeleton is not solid throughout. It is porous, filled with tiny channels and cavities. Water flows through these spaces, carrying oxygen and nutrients to the polyp's tissues, even where they extend into the skeleton. The porosity also makes the skeleton lighter than solid calcium carbonate would be, allowing corals to build large structures without the overwhelming weight that solid stone would impose. The skeleton's internal structure can be surprisingly complex. In addition to the obvious features, the corallites, theca, and septa, there are microstructural elements visible only under magnification. The aragonite crystals are organized into fibers and bundles. The orientation of these crystals affects the skeleton's strength and its ability to resist fracture. Different coral species produce different microstructures, each adapted to their particular environment and growth form. The color of coral skeletons is almost always white. Calcium carbonate itself is colorless, appearing white in reflected light. When a coral dies and its tissue decomposes, the bare skeleton that remains is stark white, a ghostly remnant of the living colony. This is why bleached corals, which have expelled their zooxanthellae but still retain their living tissue, appear white. A translucent tissue reveals the white skeleton beneath. But while the coral is alive, the skeleton is hidden. The living tissue covers it completely, creating a continuous surface across the colony. Only where the tissue has been damaged, scraped by a predator, broken by a wave, consumed by a disease, does the white skeleton show through. These exposed patches of skeleton can sometimes be recolonized by the polyp's tissue, the living layer spreading back over the bare calcium carbonate. But if the damage is extensive, the exposed skeleton may be colonized instead by algae, sponges, or other organisms. As the coral colony grows, the skeleton provides an expanding foundation. New polyps bud from existing ones, each forming its own coral light. The colony spreads outward across the substrate or upward toward the light, depending on the species and its growth form. With each generation of polyps, the skeleton grows larger. What began as a single coral light, smaller than a pencil eraser, can become a structure weighing hundreds or thousands of kilograms. The older portions of the skeleton, the corallites formed by the original polyps, may become completely buried beneath newer growth. The polyps that once occupied these corallites have long since died, replaced by their descendants. But the skeleton remains, providing structural support for the living tissue above. A large coral colony is thus a structure built by many generations. The living polyps at the surface are supported by the calcium carbonate skeletons of their ancestors. This accumulation of skeletal material over time is what builds the reef. As corals grow and die, as polyps secrete calcium carbonate throughout their lives, the total mass of skeleton on the reef increases. Dead skeleton does not disappear, it remains as a permanent structure. New corals settle on old skeletons, building upward. The reef grows thicker, rising from the seafloor toward the surface. The connection between the living tissue and the skeleton is critical to the coral's survival. The skeleton provides the framework, but it is inert. It cannot grow, cannot heal, cannot respond to the environment. The living tissue is where all the coral's biological processes occur: feeding, photosynthesis, reproduction, calcification. The tissue must remain in contact with the skeleton for the coral to function. If the tissue is stripped away by predation, disease, or bleaching-induced starvation, the skeleton remains, but the coral is effectively dead. Yet the relationship between tissue and skeleton is not entirely one way. The skeleton influences the tissue's growth form. The shape of the corallites determines how polyps are arranged. The thickness of the skeletal walls affects how much protection the polyps receive. And the overall architecture of the skeleton, branching, massive, or plate-like, dictates how the colony interacts with light, water flow, and sediment. Some coral species can modify their skeletal architecture in response to environmental conditions. Corals growing in high-energy environments with strong wave action may produce thicker, more robust skeletons than the same species growing in calm water. Corals in shaded areas may produce thinner, more plate-like structures to maximize light capture. This phenotypic plasticity, the ability of a single genotype to produce different forms depending on the environment, allows corals to optimize their growth for local conditions. The skeleton is also subject to modification after its initial formation. Bioeroding organisms, sponges, worms, mollusks, algae, bacteria, excavate tunnels and chambers within the calcium carbonate. These organisms bore into the skeleton for shelter and food. Over time, a coral skeleton can become honeycomb with tunnels, its internal structure weakened by the activities of countless small organisms. This bioerosion is a natural part of the reef ecosystem. It breaks down old coral skeleton, producing sediment, creating new habitat within the reef framework. But bioerosion also sets an upper limit on how large a coral colony can grow. If the rate of erosion exceeds the rate of calcification, the colony will eventually collapse under its own weight or be broken apart by waves. The balance between construction, the coral's secretion of new skeleton, and destruction, the activities of bioeroders and physical breakage, determines whether a reef grows or shrinks. The coral skeleton, in its accumulated mass across the reef, represents a vast reservoir of calcium carbonate. Some estimates suggest that coral reefs have deposited trillions of tons of calcium carbonate over their history. This calcium carbonate is not temporary. It can persist for millions of years. Ancient coral reefs, now exposed on land as limestone formations, contain fossil corals hundreds of millions of years old. The skeletons have been compressed and cemented into rock, but their structure remains recognizable. The process by which loose coral skeletons become solid. Limestone is called diagenesis. After a coral dies, its skeleton remains on the reef. Sediment accumulates around it. Chemical reactions, facilitated by bacteria and the chemistry of seawater, cause calcium carbonate to precipitate in the spaces between skeletal fragments. Over thousands of years, the loose material is cemented together into a solid mass. The individual corallites and skeletal structures may still be visible, but they are now part of a unified rock formation. This limestone is the ultimate legacy of the coral polyp's work. Long after the polyps have died, long after the reef itself has been buried or uplifted, the limestone remains. It records the history of coral growth, preserving information about past climates, sea levels, and environmental conditions. Scientists can drill into fossil reefs and examine the layers of coral skeleton, reading them like tree rings to reconstruct the past. The skeleton is both the means and the monument. It is the structure that allows corals to build upward toward the light, to create three-dimensional habitats, to persist across centuries. But it is also the record of that persistence, the physical evidence that corals have been building, dying, and being replaced for thousands of years. When you see a coral reef, what you are seeing is mostly skeleton. The living tissue is a thin veneer, rarely more than a few millimeters thick, covering the surface of the colony. Beneath this veneer lies meters of calcium carbonate, the accumulated skeletons of countless polyps. The reef is built not by the living, but by what the living leave behind. This is why the death of coral tissue, while tragic in the immediate sense, does not instantly destroy the reef structure. The skeleton remains. It continues to provide habitat, even if the living coral is gone. Fish still shelter in the crevices. Algae still colonize the surface. The physical structure, the three-dimensional complexity that makes the reef valuable as habitat, persists. But without living coral to add new skeleton, the reef begins a slow decline. Burrowing organisms continue their work, weakening the structure. Waves break off pieces of the dead skeleton. Storms pulverize fragile branches. Over decades, the reef erodes, transforming from a complex, three-dimensional structure into rubble and eventually into sand. What took thousands of years to build can be reduced to fragments in a human lifetime if coral growth ceases. This is the fragility hidden within the reef's apparent permanence. The skeleton endures, but only if it is continuously renewed by living coral secreting new calcium carbonate. The reef exists in a dynamic balance. Construction by living polyps must equal or exceed destruction by erosion, storms, and bioeroders. When construction stops, erosion wins. The skeleton, for all its hardness, is not indestructible. It can be broken, dissolved, worn away. Calcium carbonate is soluble in seawater, especially in cold or acidic conditions. As the ocean absorbs carbon dioxide from the atmosphere, seawater becomes more acidic, making it harder for corals to build skeletons and easier for existing skeletons to dissolve. This is ocean acidification, a quiet chemical shift that undermines the very foundation of the reef. But for now, in the clear, warm, shallow waters where coral reefs thrive, the polyps continue their work. They secrete calcium carbonate, molecule by molecule. They build corallites, theca septa. They expand their colonies, adding new polyps and new skeleton. And beneath them, the skeleton accumulates, layer upon layer, generation upon generation, year upon year. The skeleton is the silent partner in the reef story. It is not alive, but it is essential. It does not move, but it supports everything that does. It does not feed or reproduce, but it is the foundation upon which the entire ecosystem depends. And it is the skeleton, accumulated across vast areas and great spans of time, that creates the reef formations visible from space. Fringing reefs, barrier reefs, and atolls, each type built through the same process of skeletal accumulation, shaped by the geological context in which that accumulation occurs. The differences between these reef types lie not in how corals build their skeletons, but in the substrate they build upon and how that substrate changes over millennia. It is to these grand structures, these reef formations built across thousands of years, that the journey now turns. The coral skeleton accumulates. Year after year, polyps secrete calcium carbonate, colonies expand, and the reef grows. But the shape that growth takes, whether the reef hugs close to shore or forms a distant barrier, whether it encircles an island or surrounds empty water, depends not on the coral alone, but on the geological stage upon which it builds. There are 3 major types of coral reef formations. Each represents a different relationship between the living reef and the land it borders. Each tells a story about time, geology, and the patient work of coral growth keeping pace with a changing world. The most common type is the fringing reef. Fringing reefs grow directly from the shoreline, projecting seaward from the edge of the land. There is no lagoon separating the reef from the shore, no deep water channel dividing them. The reef and the land are intimately connected, the coral beginning just below the low tide line and extending outward into deeper water. Fringing reefs form when coral larvae settle on rocky shorelines in shallow tropical waters. The substrate must be hard, volcanic rock, limestone, consolidated sediment, something stable enough for larvae to attach to. Sandy beaches do not support fringing reefs because the substrate is too unstable. But where rocky shores meet warm, clear water, conditions are right. The first corals to settle establish a foothold. They grow and reproduce, their colonies expanding outward across the substrate. As these pioneering corals build their skeletons, they create new surfaces for additional larvae to settle on. The reef begins to spread, growing both horizontally across the seafloor and vertically upward toward the surface. Fringing reefs are typically narrow, extending only a few hundred meters from shore. Their width is limited by the depth at which corals can grow. Beyond a certain depth, usually around 60 meters, though most vigorous growth occurs much shallower, there is not enough light to support the zooxanthellae's photosynthesis. The reef stops growing. Its outer edge corresponds roughly to the depth limit for reef-building corals. The zone closest to shore is often called the reef flat. This is a shallow area, sometimes exposed at low tide, where hardy corals and coralline algae dominate. The water here can be very shallow, ankle-deep, or less during low tides. The corals must tolerate exposure to air, intense sunlight, and rapidly changing temperatures. Only the hardiest species survive these conditions. Moving seaward, the reef crest marks the highest point of the reef structure. This zone experiences the most intense wave action. Waves break here, dissipating their energy as they crash against the reef. The corals that grow on the crest are typically massive, boulder-shaped species with thick skeletons capable of withstanding the constant pounding. Branching corals, too fragile for such conditions, are rare or absent. Beyond the crest, the reef slope descends into deeper water. Here, wave energy is reduced. Light levels remain adequate for photosynthesis, but are less intense than at the surface. This is where coral diversity reaches its peak. Branching corals extend their delicate structures into the water. Plate corals spread horizontally. Massive corals grow in rounded domes. The reef slope is the most structurally complex zone, offering the greatest variety of microhabitats. Fringing reefs are found throughout the tropics, particularly around volcanic islands. The Hawaiian Islands, for example, are ringed by fringing reefs in many locations. So are many islands in the Caribbean, the Pacific, and the Indian Ocean. Fringing reefs represent the initial stage of reef development, the first response of corals to suitable habitat. But fringing reefs can transform into something else. Given time, thousands to millions of years, and the right geological conditions, a fringing reef can become a barrier reef. Barrier reefs also run parallel to shorelines, but they are separated from the land by a wide, deep lagoon. The lagoon can be hundreds of meters to several kilometers wide. It can be many meters deep, sometimes deep enough that corals cannot grow on its floor because light levels are too low. The transformation from fringing reef to barrier reef occurs through a combination of reef growth and geological change. If sea level rises, as it did when glaciers melted at the end of ice ages, the fringing reef grows upward to keep pace with the rising water. The coral's growth rate must match or exceed the rate of sea level rise for the reef to remain in the shallow, sunlit zone where it can thrive. Alternatively, if the island or continental shelf is subsiding, sinking slowly due to tectonic activity or the settling of volcanic islands, the effect is the same. The land's surface drops, the water deepens, but the reef continues growing upward. The corals maintain their position near the surface while the land beneath them sinks. Over time, this creates a separation. The reef remains at the surface, but the original shoreline is now submerged, drowned beneath meters of water. A lagoon forms between the reef and the current shoreline. The fringing reef has become a barrier reef. The Great Barrier Reef is the most famous example of a barrier reef system. Stretching over 2,300 kilometers along the northeast coast of Australia, it is the largest living structure on Earth. It is visible from space. Yet it is not a single reef, but a complex system of over 2,900 individual reefs and 900 islands. The Great Barrier Reef in its current form is relatively young. It began forming between 6,000 and 8,000 years ago, after the last ice age when sea levels rose and flooded the continental shelf. But the site has supported coral growth for much longer. Earlier reef structures, formed during previous warm periods, lie buried beneath the modern reef. Drilling into the reef reveals layers of coral limestone extending back hundreds of thousands of years. Other notable barrier reefs include the New Caledonian Barrier Reef in the South Pacific and the Mesoamerican Reef in the Caribbean. Each formed through the same basic process, coral growth keeping pace with rising sea levels or subsiding land, creating separation between the reef and the shore. The lagoon between a barrier reef and the shoreline is often a distinct ecosystem in its own right. The water is calmer here, protected from the full force of ocean waves by the reef crest. Sediment accumulates on the lagoon floor, fine particles of coral skeleton, shells, and terrestrial sediment washed from the land. In some lagoons, patch reefs grow, small, isolated coral formations rising from the lagoon floor, but not reaching the surface. Seagrass beds may establish themselves in the sandy areas. The lagoon becomes a nursery habitat for juvenile fish and invertebrates. But the lagoon also marks the beginning of the reef's vulnerability. The separation between reef and shore means that terrestrial influences, sediment from rivers, nutrient runoff from agriculture, pollutants from human development, can be carried into the lagoon and affect the reef. The barrier reef, while physically distant from shore, is not isolated from what happens on land. The formation of a barrier reef requires not just coral growth, but specific geological conditions. The substrate must be subsiding, or sea level must be rising, at rates that corals can match through upward growth. If the rate of change is too fast, the reef drowns, buried beneath water too deep for adequate light penetration. If the rate is too slow, or if sea level falls, the reef may be exposed to air and die. The balance is delicate. Coral growth rates for massive species range from 3 millimeters to 2 centimeters per year. Branching corals can grow faster, up to 10 centimeters per year under ideal conditions. Sea level rise at the end of the last ice age averaged about 1 meter per century over several thousand years. The corals managed to keep pace. The reef survived and grew. But if the relationship between reef growth and sea level change falls out of balance, the reef's future becomes uncertain. This is a concern now. As global temperatures rise and ice sheets melt, causing sea levels to rise at accelerating rates. Will coral reefs be able to grow upward fast enough to match 21st century sea level rise? The answer depends on many factors: the health of coral populations, the rate of calcification, the frequency of disturbances that kill corals or slow their growth. But there is a third type of reef formation, one that represents the final stage in a sequence first described by Charles Darwin in 1842. This is the atoll, a ring-shaped reef enclosing a central lagoon, with no island in the middle. Atolls are among the most distinctive features of tropical oceans. From the air, they appear as perfect circles or ovals of coral, brilliant turquoise lagoons surrounded by white sand and coral rubble, the deep blue of the open ocean beyond. They are the ghosts of islands that no longer exist. The process of atoll formation begins with a volcanic island. The island rises from the seafloor, built by repeated eruptions of lava. When the volcano emerges above the ocean surface, it becomes an island, rock and ash exposed to air, rain, and wind. If the island is in tropical waters and has suitable substrate, coral larvae settle on its submerged slopes. A fringing reef begins to form around the island's perimeter. The volcano is active for a time, perhaps thousands or millions of years, but eventually it becomes dormant. The supply of magma from the mantle ceases. No new lava reaches the surface. The volcano is extinct. Once volcanic activity stops, erosion begins to wear away the island. Rain dissolves the rock. Streams cut valleys into the slopes. The island gradually shrinks. But something else is happening simultaneously. The oceanic plate on which the volcano sits is moving. It is drifting away from the volcanic hotspot that created the island. As it moves, the plate cools. And as it cools, it contracts and becomes denser. The seafloor subsides, sinking slowly back toward the depths. The island sinks with it. But the coral reef around the island keeps growing upward. The corals maintain their position near the surface, building their skeletons upward at rates that match the subsidence. A lagoon begins to form between the sinking island and the upward growing reef. The fringing reef becomes a barrier reef. This process continues for millions of years. The island grows smaller and lower as erosion and subsidence work together. The reef grows larger and more distant from the shrinking island. The lagoon deepens and widens. Eventually, the island disappears entirely. It sinks beneath the waves, completely submerged. What remains is the coral reef, still growing, still encircling the space where the island once stood. This is an atoll. The atoll retains the approximate shape of the original island. If the island was circular, the atoll is circular. If it was elongated, the atoll reflects that shape. The lagoon marks the location of the vanished island. In some cases, the tops of the highest peaks may still protrude above the water surface as small rocky islands within the lagoon. But in most atolls, there is no trace of the original volcano. It lies beneath the lagoon floor, buried under sediment. The reef itself may emerge partially above sea level in places. Sand and coral rubble accumulate on parts of the reef platform, building up to form low islands called motifs. These islands are composed entirely of reef-derived material, coral skeleton fragments, shells, coral and algae. They are typically only a few meters above sea level, vulnerable to storms and rising seas. Darwin conceived his theory of atoll formation during the voyage of the HMS Beagle in the 1830s. He had never seen an atoll form. The process is far too slow to observe directly. But he understood the evidence. He recognized that atolls were ancient structures, the product of processes operating over geological time. His theory proposed a sequence: fringing reef around a volcanic island, subsidence of the island, transformation to barrier reef, complete submergence of the island, leaving only the atoll. At the time, Darwin's theory was controversial. There was no way to test it directly. But in the 20th century, scientists drilled into atolls to examine their internal structure. They found exactly what Darwin's theory predicted. Beneath the living coral surface lay hundreds of meters of coral limestone, layered over the basalt core of a sunken volcano. The drilling confirmed Darwin's hypothesis. Atolls are indeed the final stage in the life cycle of volcanic islands. Atolls are most common in the Pacific and Indian Oceans. The Maldives in the Indian Ocean consist almost entirely of atolls. Over 1,000 coral islands are arranged in 26 atoll formations. The Marshall Islands in the Pacific are another atoll nation. Many of the islands of French Polynesia, Micronesia, and other Pacific island groups are atolls. The lagoons of atolls can be quite large, sometimes tens of kilometers across. They are typically shallower than the open ocean beyond the reef, but can still be tens of meters deep. Some lagoons have relatively few corals growing on their floors because the water is too deep or the circulation too limited. Others have patch reefs and abundant marine life. The lagoons are often calmer than the surrounding ocean, protected by the reef ring. The three reef types, fringing, barrier, and atoll, represent stages in a continuum rather than completely separate formations. A fringing reef can become a barrier reef given the right conditions. A barrier reef can become an atoll if the island it surrounds continues to subside. The progression is not inevitable. Many fringing reefs remain fringing reefs because their substrate is stable. Many barrier reefs will never become atolls because the islands they border will not subside completely. But the potential for transformation exists, given enough time and the appropriate geological processes. All three types share fundamental characteristics. They are built by coral polyps secreting calcium carbonate. They require warm, clear, shallow water. They grow slowly, accumulating over centuries and millennia. They create complex three-dimensional structures that support diverse communities of marine life. And they are all vulnerable to changes in the environmental conditions they require. The formation of fringing reefs, barrier reefs, and atolls demonstrates the interplay between biological and geological processes. The corals provide the biological engine, growing, reproducing, secreting skeleton. But the final form of the reef depends on geology, the stability or subsidence of the substrate, the rise and fall of sea levels, the presence or absence of suitable hard surfaces for settlement. Biology and geology work together across vast spans of time to create the structures visible today. These structures, whether fringing reef, barrier reef, or atoll, are not static. They continue to grow as long as living corals cover their surfaces. They erode as waves, storms, and bioeroders break them down. They respond to changes in sea level, shifting upward or drowning, depending on the rate of change. They are dynamic formations, constantly being built and destroyed, their persistence depending on construction outpacing erosion. But the reefs exist, and have existed for thousands of years, because certain conditions have remained stable. Water temperatures have stayed within the range corals can tolerate. Light has penetrated the shallow waters. The partnership between coral and zooxanthellae has endured. When these conditions shift, when the delicate balance that sustains the reef is disrupted, the consequences ripple through the entire system. The partnership between coral and zooxanthellae has endured for more than 200 million years. It has survived ice ages, mass extinctions, and the slow drift of continents. It has adapted to changing seas, to new predators, to the gradual evolution of the ocean itself. But this partnership, for all its ancient success, exists within narrow boundaries. It requires specific conditions, temperatures within a certain range, light levels adequate but not excessive, water chemistry that favors calcification. When these conditions shift beyond what the partnership can tolerate, the relationship breaks down. This breakdown has a name. It is called coral bleaching. Bleaching is not a disease, though it may appear catastrophic. It is a stress response, a physiological reaction to environmental conditions that have moved outside the acceptable range. When a coral becomes severely stressed, it expels the zooxanthellae living within its tissues. The algae are ejected from the gastrodermal cells, released into the water column. What remains is the translucent coral polyp, its tissues now empty of the colored algae that once filled them. Without the algae, the coral's true appearance is revealed. The polyp tissue is nearly transparent. Through it, the white calcium carbonate skeleton shows clearly. The coral appears bleached, as if it has been drained of color. Reefs that were vibrant with browns, greens, and yellows become ghostly white. From above, the water that once shimmered with life looks pale and lifeless. But the coral is not yet dead. Bleached coral is stressed coral, weakened coral, but it can still be alive. The polyps remain. They can still extend their tentacles at night to capture prey. They can still secrete mucus, still respond to stimuli. The tissue persists. What the coral has lost is its primary energy source. Without zooxanthellae, photosynthesis stops. The sugars that once fueled the coral's metabolism, that powered calcification and growth, are no longer being produced. The coral must rely entirely on heterotrophic feeding, on the zooplankton it can capture with its tentacles. For most reef-building corals, this is not enough. The energy obtained from captured prey cannot match what photosynthesis provided. The coral begins to starve. The most common trigger for coral bleaching is elevated water temperature. Reef-building corals thrive within a narrow temperature range, typically between 23 and 29 degrees Celsius. But when water temperatures rise just 1 or 2 degrees above the normal summer maximum and remain elevated for several weeks, corals begin to experience thermal stress. The stress manifests first at the cellular level. The zooxanthellae's photosynthetic machinery becomes disrupted. Under normal conditions, photosynthesis produces oxygen and organic compounds. But under thermal stress, the process becomes inefficient. The algae begin producing reactive oxygen species, highly reactive molecules that include superoxide radicals and hydrogen peroxide. These molecules are toxic. They damage cell membranes, proteins, and DNA. The coral cells are injured by the reactive oxygen species produced by their own symbionts. The partnership that sustained the coral has become a liability. In response, the coral takes action. It expels the zooxanthellae. The exact mechanism is not fully understood, whether the coral actively ejects the algae, whether the algae escape on their own, or whether the symbiotic relationship simply collapses. What is clear is that the algae leave. This expulsion may be protective. By removing the source of toxic reactive oxygen species, the coral may be saving itself from further cellular damage. It is a drastic response, but possibly necessary for short-term survival. The problem is that the coral cannot survive long without its algae. Days or weeks, perhaps. Some corals can persist for months in a bleached state, surviving on captured prey alone. But if temperatures remain elevated, if the zooxanthellae do not return, the coral will eventually die. Temperature is not the only stress that can cause bleaching. Excessive light, intense solar radiation, particularly ultraviolet wavelengths, can damage both the coral and its zooxanthellae. When combined with high temperatures, bright sunlight exacerbates thermal stress. The zooxanthellae's chlorophyll, which normally captures light for photosynthesis, can absorb too much energy under intense light conditions, leading to the production of harmful reactive oxygen species. Pollution can also trigger bleaching. Chemicals in the water, pesticides, herbicides, heavy metals, even some sunscreen ingredients can stress corals. Nutrient pollution, particularly excess nitrogen and phosphorus from agricultural runoff or sewage, can disrupt the coral-algae relationship. High nutrient levels can cause the zooxanthellae to proliferate beyond the coral's ability to control them, or they can shift the balance in favor of fleshy algae that compete with coral. Changes in salinity, sudden influxes of freshwater from heavy rainfall or river discharge, can stress corals and lead to bleaching. So can extreme low tides that expose corals to air and intense heat. Even prolonged cloudy weather that reduces light levels can stress corals accustomed to bright sunlight. But among all these stressors, elevated temperature is the most widespread and the most devastating. And as global temperatures rise, mass coral bleaching events have become more frequent and more severe. Mass bleaching events occur when large areas of reef bleach simultaneously, driven by widespread environmental stress. The first documented global mass bleaching event occurred in 1998, when unusually warm ocean temperatures affected reefs throughout the tropics. Coral reefs in the Indian Ocean, the Pacific, and the Caribbean all experience bleaching. Some reefs lost more than 90% of their living coral cover. Since then, mass bleaching events have become more common. The years 2010, 2014 to 2017, and 2020 all saw significant bleaching across multiple reef systems. The Great Barrier Reef experienced unprecedented back-to-back bleaching in 2016 and 2017. Corals that survived the first event had little time to recover before being stressed again. Mortality was extensive. The pattern is clear. Ocean temperatures are rising. Heat waves in the ocean, periods of unusually warm water lasting weeks or months, are becoming more frequent and more intense. Corals that evolved over millions of years to thrive within a narrow temperature range are now regularly experiencing conditions beyond their tolerance. When a coral bleaches, its fate depends on how quickly conditions return to normal. If the stressor is removed, if water temperatures drop back to normal levels within a few weeks, the coral may recover. New zooxanthellae can enter the coral's tissues. They may come from the water column, free-living algae that settle in the vacant gastrodermal cells. Or they may come from a reservoir of algae that remain within the coral, a small population that survived the initial expulsion. Once zooxanthellae are re-established, photosynthesis resumes. The coral begins receiving energy again. Color returns as the algal populations rebuild. The tissue thickens. The coral begins to grow again. Within months, recovered coral may appear nearly normal. But recovery comes at a cost. The energy the coral used to survive the bleaching event, drawing down its stored reserves, maintaining its tissues without photosynthetic input, cannot be used for growth or reproduction. Corals that have bleached and recovered may not reproduce for a year or more. Their growth rates may be reduced. They may be more susceptible to disease. The coral has survived, but it is weakened. And if the coral bleaches again before fully recovering, the cumulative stress can be lethal. Each bleaching event depletes the coral's reserves further. Each recovery period requires energy that the coral may not have. After multiple bleaching events in quick succession, corals that might have survived a single event cannot survive the second or third. If the stress does not abate, if temperatures remain elevated for too long, the coral dies. The tissue degrades, sloughing off the skeleton. Bacteria colonize the dead tissue. Within days or weeks, all that remains is the bare white skeleton. The coral is gone. The skeleton may persist for years, a ghostly remnant of what once lived. But without living tissue to add new calcium carbonate, the skeleton begins to erode. Algae colonize the surface. Bioeroders tunnel into the structure. Storms break off branches and topple boulders. Over time, the dead reef crumbles. The ecological consequences of mass coral mortality extend far beyond the corals themselves. Fish that depend on living coral for food or shelter lose their habitat. Some species of butterfly fish feed exclusively on coral polyps. When the coral dies, these fish starve. Other fish that shelter in coral branches or defend territories on coral colonies must relocate or perish. The diversity and abundance of fish decline. Invertebrates that live within the reef structure, crabs, shrimp, worms, mollusks, lose their homes. The complex three-dimensional habitat created by living coral is gradually lost as the dead skeleton erodes. The reef becomes flatter, simpler, less hospitable to the myriad species that once thrived there. Algae often takes advantage of the vacant space. Fleshy macroalgae can grow rapidly on dead coral skeletons, particularly if herbivore populations are reduced or if nutrient levels are high. In some cases, a coral-dominated reef can shift to an algae-dominated state. The algae prevent coral larvae from settling, blocking the reef's recovery. Without intervention, protecting herbivores that graze the algae, reducing nutrient pollution, the reef may remain in this degraded state indefinitely. The transformation is not instantaneous. A reef does not shift from vibrant ecosystem to barren wasteland overnight. The process unfolds over months and years. First, the coral bleaches. Then, if recovery does not occur, the coral dies. The skeleton erodes. Algae colonizes. Fish and invertebrate populations decline. The structural complexity degrades. Each step follows from the last, a cascading series of changes initiated by the loss of the coral. But the story is not entirely one of decline. Some corals are more resilient than others. Some species can tolerate higher temperatures or recover more quickly from bleaching. Some individual corals, even within a bleached reef, retain their zooxanthellae and survive when neighboring corals die. The variation in coral responses to thermal stress has captured the attention of scientists seeking to understand resilience. Why do some corals bleach while others, exposed to the same conditions, do not? Why do some corals recover quickly while others languish? The answers lie partly in genetics, partly in the specific species of zooxanthellae the coral hosts, and partly in the coral's recent history of stress exposure. Different species of zooxanthellae have different thermal tolerances. Some species can photosynthesize effectively at higher temperatures than others. Corals hosting thermally tolerant zooxanthellae may be better able to withstand heat stress without bleaching. This has led to research into symbiont shuffling, the possibility that corals can change the dominant species of zooxanthellae in their tissues in response to environmental conditions. Under normal conditions, a coral may host one dominant species of zooxanthellae. But within its tissues, there may also be low numbers of other species, a background community of symbionts. If the coral bleaches and then recovers, the dominant species that recolonizes the tissue may be different from the one that was expelled. If the new dominant species is more thermally tolerant, the coral may be better equipped to handle future heat stress. This flexibility is limited. Symbiont shuffling is not a panacea. The different species of zooxanthellae are not all equally effective partners. More thermally tolerant species may transfer less energy to the coral under normal conditions, making the coral grow more slowly. There are trade-offs. A coral hosting a heat-tolerant symbiont may survive higher temperatures, but it may grow and reproduce less effectively when temperatures are normal. Some corals can acclimate to warmer temperatures through repeated exposure to sublethal heat stress. Corals that experience mild warming without bleaching may develop increased tolerance to subsequent heat events. This thermal conditioning is not genetic adaptation. It does not involve changes to the coral's DNA that are passed to offspring. It is a physiological adjustment, a change in how the coral and its symbionts respond to temperature. The exact mechanisms are still being studied, but they may involve changes in the production of heat shock proteins or antioxidants that protect cells from thermal damage. But acclimation has limits. A coral can adjust its thermal tolerance by perhaps 1 or 2 degrees. If temperatures rise beyond this range, the coral will still bleach. And acclimation is temporary. If temperatures return to normal for an extended period, the coral may lose its enhanced tolerance. The fundamental challenge is that the rate of environmental change exceeds the rate at which corals can adapt. Evolution through natural selection requires generations. Corals reproduce slowly. Some species spawn only once per year. Their generation time can be years or even decades for long-lived species. Evolutionary adaptation to rapidly warming oceans would require selective pressure, the survival of heat-tolerant individuals, and the death of susceptible ones, playing out over many generations. This is happening. Some corals are adapting, but the pace of adaptation may not be fast enough to keep up with the pace of warming. If temperatures continue to rise at current rates, corals may not have time to evolve the tolerance they need to survive. This is the precariousness of the coral reef in the modern ocean. The partnership between coral and zooxanthellae, refined over 200 million years, is being tested by conditions that are changing faster than evolution can respond. The balance that sustained the reef, the delicate equilibrium between coral growth and erosion, between energy input from photosynthesis and energy demands of metabolism, between the coral's needs and the algae's capabilities is shifting. When the balance shifts too far, the partnership breaks. The coral bleaches. And if the balance does not return quickly enough, the coral dies. The reef, built over thousands of years by the patient work of billions of polyps, begins to crumble. Yet even in the face of these challenges, coral reefs persist. Some corals survive where others die. Some reefs recover when conditions improve. The resilience is not infinite, but it is real. And within that resilience, within the variation in how different corals respond to stress, there may be pathways forward. The reef's future depends on whether the balance can be restored, whether the stressors that cause bleaching can be reduced, whether the conditions that allow coral and zooxanthellae to thrive can be maintained. The reef is fragile, but it is not yet lost. The polyps continue their work, building, feeding, photosynthesizing. The partnership endures, tested but not yet broken. And in some corals and some reefs, there are signs of adaptation, of resilience, of the possibility that this ancient partnership might find new ways to persist in a changing ocean. In the aftermath of bleaching, when the water is cooled and the stress is past, some corals begin to heal. The process is not dramatic. There is no sudden burst of color, no instant recovery. It unfolds slowly, over weeks and months, as quietly as the bleaching itself occurred. The first sign is subtle. The bleached coral, stark white against the reef, begins to show hints of color. Faint browns and greens appear in the tissue. These are zooxanthellae, returning to the empty gastrodermal cells. They may come from the water column, free-living algae that settle in the vacant spaces where their predecessors once lived. Or they may come from a small reservoir that remained within the coral, a population that survived the expulsion and now begins to multiply. As the zooxanthellae re-establish themselves, photosynthesis resumes. The coral begins receiving energy again. The sugars produced by the algae flow into the coral's tissues, fueling metabolism, supporting cellular repair. The coral, which has been surviving on the meager energy obtained from captured prey, can now breathe easier. The partnership, broken by stress, begins to reform. Over the following months, if conditions remain favorable, the coral's color deepens. The population of zooxanthellae grows. The tissue thickens slightly as the coral rebuilds its cellular reserves. Chlorophyll concentrations increase. The coral looks healthier, more vibrant. To an observer, it may appear that the coral has fully recovered. But recovery is more complex than it appears. The coral may look normal, yet beneath the surface, the effects of bleaching persist. The energy the coral expended to survive the bleaching event, drawing down its stored reserves of lipids, proteins, and carbohydrates, must be replenished. This replenishment takes time. Energy that would normally go toward growth or reproduction is instead diverted to rebuilding reserves. Studies of Caribbean corals recovering from bleaching have revealed that different tissues recover at different rates. Chlorophyll levels and zooxanthellae densities may return to normal within weeks or a few months. But lipid reserves, the primary energy storage in many coral species, can take a year or longer to fully recover. Protein and carbohydrate reserves follow their own timelines, depending on the species. Calcification, the process by which the coral builds its skeleton, is often impaired during and after bleaching. Some corals stop calcifying entirely when bleached, and in severe cases, the skeleton may actually dissolve, losing more calcium carbonate than is being deposited. When conditions improve and the coral begins to recover, calcification resumes, but growth rates may remain lower than normal for months. The coral is alive, but it is not thriving. Reproduction is another casualty of bleaching. Corals that have recently bleached often fail to reproduce during the next spawning season. The energy required to produce eggs and sperm is substantial, and a coral recovering from bleaching simply does not have the reserves to invest in reproduction. The coral prioritizes survival over reproduction, a biological calculation that makes sense in the short term but has long-term consequences for reef recovery. This means that even when a coral survives bleaching and appears to recover, its contribution to the next generation may be lost. If bleaching events occur frequently, every few years rather than every few decades, corals may spend most of their time recovering rather than reproducing. The population cannot replenish itself. Slowly, the reef declines not through mass mortality, but through the gradual erosion of reproductive capacity. The time required for full recovery varies by species and by the severity of the bleaching. Some corals can recover within months. Others require a year or more. But recovery is only possible if conditions return to normal and remain stable. If the coral experiences another bleaching event before fully recovering, the cumulative stress can be lethal. Yet not all corals respond to bleaching in the same way. On a reef where many corals have bleached, some individuals remain unbleached. Among those that do bleach, some die quickly while others linger, and some recover fully. This variation is not random. It reflects differences in physiology, genetics, and the specific partnerships each coral has formed with its zooxanthellae. The zooxanthellae living inside coral tissues are not a single species. They are a diverse group of dinoflagellate algae with many different genetic types within the genus Symbiodinium and related genera. Each type has its own characteristics, its own growth rate, its own photosynthetic efficiency, its own tolerance to temperature and light. Some types of zooxanthellae are better adapted to high temperatures than others. They can maintain photosynthetic function at temperatures that would cause other types to malfunction and produce toxic reactive oxygen species. Corals hosting these thermally tolerant zooxanthellae are less likely to bleach during heat stress. They can withstand higher temperatures without expelling their symbionts. This has led to the recognition that the coral-algae partnership is not fixed. A coral does not necessarily host the same species of zooxanthellae throughout its life. In some cases, the dominant symbiont type can change in response to environmental conditions. This process is called symbiont shuffling. Under normal conditions, a coral may host one dominant species of zooxanthellae that provides most of the photosynthetic energy. But within the coral's tissues, there may also be low numbers of other species, a background community that persists at low densities. If the coral bleaches, it expels most or all of its zooxanthellae. When the coral recovers and zooxanthellae recolonize the tissue, the dominant species that returns may be different from the one that was expelled. If the new dominant species is more thermally tolerant, the coral may be better equipped to handle future heat stress. The coral has, in effect, changed partners, swapping a heat-sensitive symbiont for a more resilient one. This flexibility provides a potential mechanism for corals to adapt to warming oceans without requiring genetic changes in the coral itself. But symbiont shuffling is not a perfect solution. The different species of zooxanthellae are not all equally effective partners. Some species that are more tolerant of high temperatures are less efficient at transferring energy to the coral under normal conditions. A coral hosting a heat-tolerant symbiont may grow more slowly than a coral hosting a more productive but less heat-tolerant symbiont. There are trade-offs. This creates a dilemma for the coral. In a stable, cool environment, the most productive symbiont provides the best partnership. But in a warming ocean with frequent heat stress, the most heat-tolerant symbiont offers the best chance of survival, even if it means slower growth. The optimal strategy depends on what the future holds, a future the coral cannot predict. Some corals appear capable of acclimating to warmer temperatures through repeated exposure to sublethal heat stress. Corals that experience mild warming without bleaching may develop increased tolerance to subsequent heat events. This is not genetic adaptation. It does not involve changes to the coral's DNA that are passed to offspring. It is a physiological adjustment, a change in how the coral and its symbionts respond to temperature. The mechanisms underlying thermal acclimation are still being studied. They may involve the production of heat shock proteins, molecular chaperones that help protect cellular proteins from damage at high temperatures. They may involve increased production of antioxidants that neutralize the reactive oxygen species produced during heat stress. They may involve changes in the coral's metabolism or in the photosynthetic machinery of the zooxanthellae. Whatever the mechanisms, the result is that some corals can increase their thermal tolerance by 1 or 2 degrees Celsius through acclimation. This is significant. It could mean the difference between bleaching and surviving during a moderate heat event. But acclimation has limits. If temperatures rise beyond the acclimated range, the coral will still bleach. And acclimation is not permanent. If temperatures return to normal for an extended period, the coral may lose its enhanced tolerance. Energy reserves also play a critical role in determining whether a coral survives bleaching and how quickly it recovers. Corals with high levels of stored energy, particularly lipids, are better able to withstand the energy deficit that occurs during bleaching. They can survive longer without photosynthetic input. They can recover more quickly once zooxanthellae return. Different coral species store energy in different forms. Some rely heavily on lipids. Others store more protein or carbohydrate. The specific energy reserve that a coral draws upon during bleaching is species-specific. But the general principle holds. Corals with larger energy reserves are more resilient to bleaching. This means that anything that affects a coral's ability to build energy reserves also affects its resilience. Corals that have been stressed by other factors, disease, predation, pollution, sedimentation, may have depleted reserves and be more vulnerable to bleaching. Corals in poor health entering a heat event are less likely to survive than corals in good health. The existence of variation in coral responses to heat stress, some corals surviving where others die, some recovering where others do not, provides a foundation for evolutionary adaptation. If heat-tolerant individuals survive and reproduce while heat-sensitive individuals die, the next generation will contain a higher proportion of heat-tolerant genes. Over many generations, the population can evolve increased thermal tolerance. This process is already happening. Scientists have documented genetic differences between coral populations from warmer and cooler locations. Corals from naturally warmer environments often show greater thermal tolerance than corals of the same species from cooler locations. Some of this difference is acclimation, but some appears to be genetic, heritable differences in thermal tolerance that have evolved through natural selection. The challenge is time. Evolutionary adaptation through natural selection requires generations. Corals reproduce slowly. Many species spawn only once per year. Their generation time can be years or decades. For selection to produce substantial changes in thermal tolerance requires many generations, which means many decades or even centuries. Ocean temperatures are rising rapidly. The pace of warming in recent decades is faster than at any time in coral evolutionary history, except during periods of mass extinction. The question is whether corals can adapt fast enough to keep pace with the rate of change. The answer is uncertain. Some coral populations may be able to adapt. Those with high genetic diversity, large population sizes, and short generation times have the best chance. But many coral species do not meet these criteria. They reproduce infrequently, their populations are fragmented, and genetic diversity is low. For these species, evolutionary adaptation may be too slow. There is also the issue of how much adaptation is possible. Thermal tolerance is not infinitely flexible. There are physiological limits to how much heat a coral and its zooxanthellae can withstand. The chemical reactions of metabolism and photosynthesis have optimal temperature ranges. Push beyond those ranges, and the reactions begin to fail. No amount of evolutionary adaptation can overcome fundamental biochemical constraints. Yet even within these constraints, there is hope. Some coral reefs have shown remarkable resilience. The reef at Palmyra Atoll in the Central Pacific experienced severe bleaching in 2015 and 2016, losing much of its living coral cover. Scientists expected the reef would take decades to recover, if it recovered at all. But within just a few years, coral cover had rebounded significantly. The reef recovered far faster than predicted. The recovery at Palmyra was aided by the reef's remoteness. The atoll is far from human populations, far from pollution, overfishing, and coastal development. The local stressors that degrade many reefs were absent. When the thermal stress passed, the corals could focus all their energy on recovery rather than fighting multiple simultaneous stressors. This demonstrates that reducing local stressors can enhance reef resilience, giving corals a better chance of surviving and recovering from bleaching events. Other examples of resilience have been documented across the tropics. Corals growing in naturally warm or variable environments, mangrove lagoons, shallow pools with extreme daily temperature swings, show greater thermal tolerance than corals from stable environments. These supercorals, as some researchers call them, may hold keys to understanding how corals can survive in a warming ocean. The idea has emerged that these resilient corals could be transplanted to degraded reefs, seeding them with heat-tolerant genotypes, or that reefs could be managed to promote the survival and reproduction of the most resilient individuals. These are not solutions to the underlying problem of warming oceans, but they may buy time, allowing some reefs to persist while broader efforts to address climate change take effect. The coral reef in the modern ocean exists in a state of precarious balance. The partnership between coral and zooxanthellae continues, tested by conditions that are changing faster than evolution typically allows. Yet the partnership has not broken entirely. Some corals bleach and recover. Some shift their symbionts to more heat-tolerant types. Some acclimate to warmer conditions. Some appear to possess genetic variants that confer greater thermal tolerance. The future of coral reefs depends on whether these mechanisms of resilience and adaptation can operate fast enough and broadly enough to allow corals to keep pace with a changing climate. It depends on whether the stressors that weaken corals can be reduced. And it depends on whether the conditions corals need, warm but not too warm, clear water, adequate light, stable chemistry, can be maintained or restored. The reef is fragile, but it is not yet lost. The polyps continue their work. The partnership between coral and algae endures in millions of colonies across the world's tropical oceans. And in the variation among those colonies, in the differences in how they respond to stress, there exists the raw material for adaptation. Not a guarantee of survival, but a possibility. A pathway through which this ancient partnership might persist, transformed but unbroken, into an uncertain future. There is a kind of intelligence that has nothing to do with thought. It does not require a brain, does not involve planning or consciousness. Yet it is profound. It is the intelligence of systems that work, of relationships that endure, of solutions discovered not through intention, but through the patient filtering of time. The coral reef possesses this intelligence. Consider what the reef accomplishes. In nutrient-poor waters where life should be scarce, it creates abundance. From sunlight and dissolved minerals, it builds structures visible from space. It shelters a quarter of all marine species on less than 1% of the ocean floor. It transforms carbon dioxide into calcium carbonate, storing it for millennia. It protects coastlines from storms, feeds millions of people, and generates billions of dollars in economic value. None of this was designed. None of it was planned. Yet it works. The genius lies in the partnerships. A coral polyp, no larger than a grain of rice, cannot build a reef alone. It does not have the energy. But the polyp does not work alone. Inside its tissues live billions of microscopic algae. The algae photosynthesize, capturing sunlight and converting it to sugar. They give up to 90% of what they produce to the coral. In return, the coral provides shelter, protection from predators, and waste products the algae need as nutrients. Neither organism could build a reef independently. Together, they accomplish what seems impossible. This is the first wisdom of the reef, that cooperation can achieve what competition cannot. The polyp and the algae are not competitors. They are partners. Their success is shared. When one thrives, both thrive. When the partnership breaks, both suffer. The reef exists because billions of these partnerships have held, have endured, have passed the test of time. But the partnerships extend beyond the polyp and its algae. The reef itself is a network of relationships. Cleaner fish remove parasites from larger fish. Clownfish shelter in anemones while defending them from predators. Gobies and shrimp share burrows, one providing security, while the other digs. Coral and algae cement loose rubble, stabilizing the structure that corals build. Parrotfish graze on algae that would otherwise overgrow coral, while producing sand that builds beaches. Everywhere you look, organisms are connected through feeding, through symbiosis, through the simple fact of living in proximity. The reef demonstrates that complexity emerges from connection. No single organism controls the reef. No species directs the others. Yet the system functions. Energy flows from producers to herbivores to carnivores. Nutrients cycle from living organisms to decomposers and back to producers. The structure grows as corals build and erodes as bioeroders consume, the balance determining whether the reef expands or contracts. It is messy, chaotic, constantly changing, yet somehow stable. This is the second wisdom. That order can emerge without central control. The reef is not organized from the top down. It organizes itself. Each organism responds to local conditions, to what it senses immediately around it. The polyp extends its tentacles when it detects prey. The zooxanthellae photosynthesize when light is available. The fish feed where food is abundant. No one coordinates these actions. Yet collectively, they create an ecosystem that persists for thousands of years. The reef also teaches patience. A coral colony growing at 2 centimeters per year needs 500 years to reach 10 meters. The Great Barrier Reef has been forming for hundreds of thousands of years across multiple cycles of growth and retreat. The limestone beneath modern reefs contains the compressed skeletons of corals that lived millions of years ago. Nothing about the reef happens quickly. The polyps build slowly, molecule by molecule. The larvae drift for days or weeks before finding a place to settle. The recovery from bleaching takes months or years. The evolution of thermal tolerance may take centuries. Yet this slowness is not weakness. It is the pace of processes that endure. Fast growth can be fragile. Branching corals grow quickly, but break easily. Slow growth can be robust. Massive corals add only a few millimeters per year, but they can survive for 1,000 years. The reef's longevity comes not from speed, but from persistence. The polyps keep building. The partnerships keep forming. The larvae keep settling. Day after day, year after year, century after century. This is the third wisdom, that time transforms the small into the monumental. A single polyp secreting a few milligrams of calcium carbonate seems insignificant, but multiply that polyp by billions, that day by millennia, and you build islands. You create structures that alter ocean currents, that protect entire coastlines, that support ecosystems rivaling rainforests in their diversity. Small acts, repeated consistently over long periods, accumulate into something extraordinary. The reef also embodies resilience. It is damaged repeatedly. Storms break coral branches. Crown-of-thorns sea stars consume living tissue. Bleaching events kill corals across vast areas. Disease sweeps through populations. Yet the reef persists. Broken fragments can regenerate into new colonies. Coral larvae settle on dead skeleton and begin building again. Surviving corals reproduce, their offspring repopulating the reef. Recovery is slow, often incomplete, but it happens. This resilience has limits. If disturbances occur too frequently, if corals are not given time to recover between events, the system can collapse. If the fundamental conditions that support reefs, the water temperature, the water chemistry, the availability of light shift beyond what corals can tolerate, no amount of resilience will save them. The reef is robust within the range of conditions it evolved to handle, but that range is not infinite. Yet even facing unprecedented challenges, the reef demonstrates its capacity to respond. Some corals bleach while others do not. Some recover quickly, others slowly, and the variation is not random. It reflects differences in genetics, in the species of zooxanthellae hosted, and prior exposure to stress. This variation is the raw material for adaptation. The corals that survive pass their genes to the next generation. Over time, the population shifts toward greater heat tolerance. It is evolution in action, natural selection operating in real time. The question is whether this adaptation can happen fast enough. The oceans are warming rapidly. The pace of change exceeds anything in recent coral evolutionary history. Some coral populations may adapt, others may not. The reef's future remains uncertain. But uncertainty is not the same as hopelessness. The reef at Palmyra recovered faster than anyone predicted. Supercorals in naturally warm environments show that thermal tolerance is possible. Efforts to reduce local stressors, overfishing, pollution, coastal development, have demonstrably improved reef resilience. The tools exist to help reefs survive. What is needed is the will to use them. This is the fourth wisdom, that resilience depends on both the capacity to recover and the space to do so. A coral can heal from injury if given time. A reef can recover from bleaching if conditions stabilize. But if the injuries keep coming, if the stressors never relent, even the most resilient system will eventually fail. Protecting the reef means not just protecting the corals themselves, but protecting the conditions they need to persist. The reef also holds lessons about beauty emerging from necessity. The vibrant colors of healthy coral come from the algae that provide energy. The complex three-dimensional structure that makes reefs visually striking is the same structure that provides habitat for thousands of species. The diversity of forms, branching, massive, plate-like, represents different solutions to the challenges of capturing light, withstanding waves, and competing for space. Every aspect of the reef that humans find beautiful serves a functional purpose. Beauty and function are not separate. They are intertwined. Perhaps this is why coral reefs captivate us. They are alien animals that build stone, partnerships between kingdoms of life separated by billions of years of evolution, ecosystems that thrive in barren seas. Yet they are also deeply familiar. The reef demonstrates principles we recognize: cooperation, persistence, adaptation, the emergence of complexity from simple rules repeated many times. The reef is both strange and intuitive, both fragile and enduring. Standing at the edge of a reef, floating above one with mask and fins, you witness something that has been unfolding for hundreds of millions of years. The polyps, extending their tentacles tonight, are engaging in the same behavior their ancestors performed in ancient seas when the continents were arranged differently and most modern species did not yet exist. The partnership between coral and zooxanthellae predates the dinosaurs. The process of calcification that builds the reef today is the same process that built reefs during the Triassic, the Jurassic, the Cretaceous. You are witnessing continuity across deep time. Not because the reef is unchanging, it is constantly changing, but because the fundamental processes persist. Polyps still capture prey. Algae still photosynthesize. Calcium carbonate still accumulates. Larvae still settle and begin the cycle again. The reef adapts, evolves, shifts in composition and structure, but it continues. This is the fifth wisdom, that persistence does not require perfection. The reef has been damaged countless times in its history. It has survived mass extinctions, ice ages, sea level changes of more than 100 meters. Each time it recovered, though the corals that rebuilt may have been different species than those that died. The reef as a phenomenon persists even when individual reefs, individual corals, individual polyps do not. The quiet genius of the coral reef lies in its capacity to create abundance from scarcity, complexity from simplicity, permanence from transience. A polyp lives for months or years. The skeleton it builds can persist for millennia. The calcium carbonate deposited today may one day be compressed into limestone, uplifted by tectonic forces and exposed as mountains. The polyp does not know this. It has no awareness of the future. It simply builds. Yet in that building, it participates in processes that extend far beyond its own existence. This is perhaps the deepest lesson the reef offers, that significance can emerge from actions that have no inherent knowledge of their significance. The polyp secreting calcium carbonate is not trying to build a reef. It is responding to immediate biochemical signals, the presence of calcium and carbonate ions, the energy provided by its zooxanthellae. Yet the cumulative result of millions of polyps responding to these same signals is an ecosystem that supports vast biodiversity, protects coastlines, and sustains human communities. The reef demonstrates that purpose can be found in process. The polyp's purpose, if such a word can be applied to an animal with no brain, is to survive and reproduce. Everything it does serves those goals. But in serving those goals, it creates something larger than itself. The reef is not the polyp's intention. It is the consequence of the polyp's existence, multiplied across space and time. In this way, the reef is a metaphor, though it is not trying to be. It simply is a living system built through cooperation, sustained through persistence, maintained through the constant work of organisms responding to their environment. It shows what can emerge when simple processes operate consistently over long periods. It reveals how complexity can arise without a blueprint, how order can emerge from apparent chaos, how beauty and function can be inseparable. The reef also reminds us that everything is connected. The polyp depends on the algae. The algae depend on sunlight and nutrients from the polyp. The fish depend on the reef structure for shelter. The reef structure depends on the polyps continuing to build. The polyp's ability to build depends on the energy from algae. Remove any link in this chain and the entire system is affected. The reef is not a collection of independent parts. It is a web of interdependencies. When you understand this, you understand why the reef is fragile. Not because any single component is weak, but because the system depends on relationships remaining intact. When the coral-zooxanthellae partnership breaks through bleaching, the coral loses its primary energy source. When corals die, the fish lose their habitat. When herbivores disappear, algae overgrows the reef. The strength of the system lies in its connections. Its vulnerability lies there too. Yet the reef also shows that relationships can be renewed. Bleached corals can reacquire zooxanthellae. Dead reefs can be recolonized by new larvae. Partnerships can reform. The capacity for recovery exists as long as the fundamental conditions that support life remain within tolerable bounds. Tonight, as you rest, coral polyps across the world's tropical oceans are extending their tentacles. They are capturing zooplankton from the water. Inside their tissues, zooxanthellae are waiting for sunrise, when photosynthesis will resume. Beneath the polyps, calcium carbonate is accumulating, adding infinitesimal layers to structures that may outlast human civilization. Larvae are drifting in the currents, searching for places to settle. Fish are sheltering in coral branches, safe from predators. The reef continues its work. It does not know that it is teaching us. It does not know that it is demonstrating principles of cooperation, persistence, resilience, and interconnection. It simply exists, doing what it has done for hundreds of millions of years, building, adapting, persisting. The quiet genius of the coral reef is that it accomplishes so much while intending nothing. It is intelligence without consciousness, wisdom without thought, purpose without planning. And perhaps that is the final lesson, that meaning can be found not in grand intentions but in simple acts repeated faithfully. The polyp builds. The algae photosynthesize. The larvae settle. The partnerships form. And from these small repeated acts emerges something magnificent, a living city beneath the waves, a foundation for abundance, a testament to the power of collaboration across deep time. The reef is fragile, yes. It faces challenges unprecedented in its history. But it is not yet lost. The polyps continue. The partnerships endure. And in that continuation, in that endurance, there is hope. Not the hope of certainty, but the hope of possibility. The possibility that this ancient system, tested by time, will find ways to persist in a changing world. The possibility that the quiet genius of the reef, its capacity to create, to adapt, to endure, will prove sufficient for whatever comes next. This is the end of your journey.